first sparks on quiet streets, two hearts finding where to meet. But life pulled us apart, left me talking to the dark. If I called you tonight, would it feel the same? Or have we both moved on to different names? Take me back to the start, before the quiet and the scars, when the world felt small and your hands felt warm. Take me back to the start, if only for a moment. I've been up since 3, scrolling back where we used to be. Half truths in this late night chat, both of us know.
Hello. All right, we got biology paper 2 to prepare for. So yeah, this is a live stream for biology paper 2, and yeah, let's get straight into it. So what I'm going to do is go through content and exam questions. So let's go. Woo! So what this is supposed to be, my AI image, is of a nervous system. Now, whether you're doing combined or triple, I will point out when different topics are combined or triple, but I'm covering everything, so triple and combined for biology, for AQA. So this is the nervous system. I'm going to talk about the nervous system. So let's get into it. This is part of Unit 5. So really you're going to be doing like the nervous system. For the next hour, I'm going to be talking about the nervous system, the menstrual cycle, hormones, control of blood glucose, control of temperature, plant hormones if you're doing triple, and that's the first unit, and the ruler drop test. And then in the second part of this live, I'm going to be doing, or not the second part, the next unit is going to be DNA, alleles, inheritance, natural selection, selective breeding, speciation, classification, that whole unit. And then for the final bit, the third part is going to be on ecology. Again, if you're doing triple, you've got quite a bit more to do, but it's basically abiotic, biotic factors, using sampling, using quadrats, adaptations, the water cycle, the carbon cycle, etc., and food security and stuff like that. If you're doing triple, you've got pyramids of biomass, and you've got the decay practical as well. All right, let's get into it. Let's get on. Let's get going. So first up, you have a nervous system. Now, your nervous system is one of your messaging systems that connects you to the outside world, and it feeds back information about what's going on about you. So let's have a little look and let's get into that. So this woman here, stimulus. A stimulus is a change in the environment, so light from an insect flying near her eye. And where would we find the receptors? Receptors are special cells that will detect a change in the environment. So if an insect is flying near this woman's eye, look at that incredible drawing, then receptor cells found in our retina are going to detect the light bouncing off the insect into her eye, and that's going to go to the brain, which is the coordinator, and the effect of all of that is going to be her muscles contract, and she blinks and protects her eye from any damage. Now we're going to get into this properly. So when we look at this, first of all, see if you can tell me what W, X and Y are. Okay, let me go through this. The main reason I put this up, I want to actually show you the whole reflex arc. So when someone touches a pin or a hot object, but this is the same thing like an insect going near your eye, we're going to have we have receptors here. Now the pin itself is a stimuli, it's a change in the environment, a stimulus, singular. Now we then have a sensory neuron. This diagram's a bit crap for this, but a sensory neuron, an electrical signal goes down this wire called a sensory neuron, and that ends up getting here into our spine. Now, what is this? If you run your finger up your neck, you will feel little bones. They're called vertebrae, and they're made of white matter and grey matter. Now, your sensory neuron will go into that, but then there is a little gap between the neurons, and then we have a relay neuron, and then we have a motor neuron. So I'm going to write the order in a second because this is super messy. So this is just part of the spine, so spinal cord, whatever we want to call that, who cares, and that is muscle. But I want to run through the order you need to know for a reflex arc. So a reflex arc, you might get, if you're lucky, a 5 marker on the reflex arc, which is like you put your hand on a hot object, you pull it away. Explain how that works. So how this works is like this. There's about 8 points. You probably need 5 to get all the marks out of 8. So stimulus. Now you have to read the question to find out what the stimulus is. And then we have receptors or receptor cells. Now you have receptor cells in your skin for detecting pain, touch and heat. Receptor cells at the back of your eye for detecting light. You have receptor cells in your ear for detecting sound. Receptor cells in your nose called olfactory receptors that detect smell. And then you have taste receptors at the back of your tongue. So you've got these 5 senses. Well, all of those contain receptor cells. Receptor cells create an electrical signal. So we want to say send an impulse. Now, an impulse is a signal, and what I want to say is a stimulus detected by, detected by receptor cells, send an impulse, and then we want to go down or along. Let's say the word along, in case you think it has to go downwards. So, along sensory neuron. Now think about it, this is the sensory nerve. If you don't like the word neuron, you're allowed to use the word nerve, okay? So you can use N-E-R-V-E instead of neuron. That sends this impulse along a sensory neuron. I'm going to come back to this. It then crosses a synapse. Oh my good gosh. It then crosses synapse. We'll come back to what a synapse is in a second. It then goes to the relay neuron. Now the relay neuron is a little wire that we have up here. If I wipe all this rubbish out, the relay neuron is a little wire here that runs through your vertebrae in your spine, but also runs through your brain. So the relay neuron is found here. This is the sensory neuron, and the impulse is going in this direction, right, through the relay neuron, and then along M here, a motor neuron. So we're going to go from relay neuron, but there is a gap between the relay and the motor, which we call a synapse. So it's going to cross another synapse, and then it's going to go motor neuron. Oh my days! And then we are going to have, it's going to go, in this situation, X is a muscle, but the muscle, it's not always a muscle it goes to. It either goes to a muscle or gland. We call these the effectors because they have the effect and you respond by pulling your arm away. In this case, anything mechanical or physical, like blink, shutting your eye, moving your arms or legs, that is going to be a muscle. Anything which then releases a chemical is going to be a gland, which is a hormone. So synapse, motor neuron, and then we're going to have effector, and in this case it's a muscle. And I'm going to show you, we're going to apply this in a 6-mark question in a second. So all of that jabroni, stimulus detected by receptor cells in your, like let's say she's touching a pin, so this would be a stimulus is the pin, that's detected by receptor cells in her skin. They create an electrical signal or impulse that goes along the S here, the sensory neuron. It crosses a gap in the neurons called a synapse, goes to the relay neuron, then it goes to the motor neuron, and then the muscle, and the effect is she pulls her arm away, the muscle contracts. Now what I want to do is just show you what happens at the old synapse. So let me go here, and then I'm going to give you a bunch of questions on this. So I'm just going to go through the content on the nervous system, which will take another 5 minutes, and then I'm going to give you a whole bunch of questions. So steady on, steady on. Now, what goes on at a synapse? A synapse is the end of one of these neurons. So remember, just to recall, there are 3 neurons, like wires. You have you have like a sensory neuron. You don't need to know why it looks like that, but you have a sensory neuron. I'm just going to put S. Then that connects to what we call a motor neuron. The relay neuron doesn't look like that. Motor neuron does look like that. You're not going to be asked what they look like, so don't worry. On your syllabus, you don't have to know what they look like. Like the relay neuron looks a bit different, looks a bit like that, but you don't really have to know. There's just 3 wires, but notice there is a gap here. So what we're going to do is investigate that gap. So at the end of the sensory neuron, so this would be the one that's connected to your skin or connected to your ear, connected to the back of your eye, your retina cells, connected in your nose and your tongue for taste. You have a sensory neuron. Then the gap is called the synapse. And then what would this neuron be? This neuron has to either be a relay or a motor. Now, sometimes there's no relay, but in this situation, this is a relay neuron. So this is a gap. So if you're asked what is a synapse, if you get that question, what is a synapse? Your answer, synapse, is a gap between 2 neurons. Gap between 2 neurons. Nice and easy. Now there's another thing you have to know. How does the message cross the gap? So I'm just going to show you. The electricity comes down here like this, because your nervous system uses electricity. When it reaches here, causes these little vesicles. You don't need to know this, but it causes chemicals. I'm just going to put them in circles like that. They're in these little sacs. These circles then move to the edge and they release their chemicals like that, and the chemicals diffuse across the gap from high to low concentration. On the other side, they lock on to special target sites here, and they create the electricity again over here in the other neuron. So the signal goes electric, chemical, and then back to electric. So the nervous system, generally we describe it as using electrical signals, but there is a little bit of chemical messaging going on there in the synapse. The gap between the neurons is chemical, and they diffuse across the gap. So if you are asked this question, how does the message cross the synapse? Excuse for the crap writing, right? We're trying to go fast, right? How does the message cross the synapse? That would be 2 marks. So I want to say as chemicals, get myself a mark, and then diffusion, get myself a mark. Now, do you have to say neurotransmitter? Not necessarily, but that is the name of those chemicals. Those ones I've drawn there are neurotransmitters. That is the specific name of that chemical, is a neurotransmitter. Oh my gosh, let me see. Okay. Right, guys, let me give you, that is the detail at the synapse. Couple of other things before we finish with this. Okay, because we don't want to be on this bit all night. Oh no, I didn't want you. Go away. Bish. Bush. Bosh. Okay, couple of other things you could be asked about. Then we're going to do some questions. Couple of other things are, you might be asked, what is a reflex? So what is a reflex? What is a reflex? 2 marks. So it's a response. When a fly goes near your eye and you blink, when you touch a hot object and you pull your hand away, it's a reflex. So we want to say, now if you can't think of the word, rather than write something that the examiner doesn't understand, so involuntary means you're not, you don't have to think about it. You can say unconscious. This is another word, but I'm not even going to write unconscious because the chances of me spelling that is slim. So involuntary, or you can say you do not, if you can't remember that word, just say you do not need to think about it. right? So it's unconscious or it's an involuntary response. We need to add another thing to this because it's 2 marks. It is rapid. Now, you don't have to say rapid, you can say fast. So if I'm on the mark scheme, involuntary response, a rapid or a fast unconscious response or involuntary response. That's what a reflex is. You don't have to think about it. All right. What else? Let's smash through and do a couple of questions on this. So here we go. 6 marker. Woman touches a hot object. The woman moves her hand away rapidly. Describe how the woman's nervous system coordinates this reflex action. See if in your head you can remember 6 of the bullet points we talked about. You don't need to get all of them, just 6 of them, about how she pulled her hand away from the match. Don't forget to hit the like while you're in here, guys.
If you push the button,
Like me.
Would you like it?
Like.
do it. Do it.
Do it.
Okay, I'm going to go through this. So I'm just going to give you some 6 bullet points I would use here. Okay, and I'm bullet pointing. So describe how the woman's nervous system coordinates the reflex action. So I'm just going to start with, I'm not bothering to write this, by the way. The flame is the stimulus, and she has receptor cells in skin. Now, if I wanted to, I could say, oh, the match is the stimulus, receptor cells in her skin detect the stimulus. That's all very nice, but I can't be bothered to write all of it. So I'm just going to say receptor cells send impulse. Now, probably I should clarify, if you've got the time, to write electrical impulse, because sometimes on the mark schemes they get deep and they're like, oh, we have to say electrical impulse or electrical signal or electrical message. If you don't want to say impulse, you can say message or signal, but receptor cells send electrical signal or electrical impulse. And what I want to say is along sensory neuron. So you've got to get the one right. Why is it the sensory neuron? Because it's connected. This wire is connected and is going in this direction towards our brain and spine. So it's connected to one of the 5 senses. So along sensory neuron. By the way, you can say nerve if you prefer. Now that would score you 2 out of 6 marks. Now I'm just have to talk about this once, and you don't have to talk about it at all probably, but I'm going to mention it. I'm going to say message crosses synapse. Now it doesn't say explain, so yes, I'd like to say from a high to low concentration by diffusion as a chemical. So message crosses synapse as chemical. I'm not going to go into too much detail, and the reason why, this is describe, not explain. So I'm not going to get into too much detail on that. That's going to get me a 3rd mark. Then I'm going to say, then goes from relay neuron. Now I could say to the spinal cord if I want, or the brain, or the coordinator. We call the brain and the spinal cord the coordinator. That's where the relay neuron runs through, and they coordinate your response. So I could add that if I want, but I'm just going to go then from relay neuron to motor neuron. Now it does cross another synapse in between these 2 neurons, but I've already said it, so the examiner's not giving me 2 marks for mentioning synapse twice. You're just not getting that extra mark for that. And then I'm going to say to the muscle. And what do muscles do? That, by the way, the muscle is an effector, so they'll probably give you the mark if you said effector. And muscle contracts. Now, they haven't asked me to say about what the response is, but if you want to know response, she pulls her arm away. So that happens after the muscles contract. So that's going to get me all the marks. I've probably put about 7 or 8 things there, and I don't need all of those to get the marks, okay? Yes, there's other stuff. I could have gone into detail about how the message crosses the synapse from a high concentration to a low by diffusion as a chemical. All right, let's have a go at this one. Describe the function of receptors in the skin. So what is it that the receptors in your skin actually do? So I've already kind of explained it. What do the receptors in your skin do? See if you can put it up on a chat. I lost my glasses, my normal glasses. I don't have them. Fuck, excuse me. What was that? Never mind. Right. Good. Detect stimulus. And if it's a 2 marker, you want to say create an impulse as well. Boom! I'll give you the proper boom at some point. Right. Yeah. So detect stimulus. And what else did we say? They detect the stimulus. What else? What else? What else? What else? Oh, I might want to say create impulse, because it's your receptor that first turns it's really a transformer, so a transducer. It transforms, so say like a match, that's heat energy. This is transforming heat energy into electrical energy, right? So it's a transformer, your receptor cells. The ones in your eye change light into electrical impulse. The ones in your tongue change chemical taste into electricity. The ones in your nose change chemicals into electricity. You get the coup, right? Pressure, when someone pokes you. No, did he? When someone pokes you, your mechanical receptors change pressure into electricity. You get the coup. Right, type of effector. There's 2 types of effector, and what do they do? So who can give me the 2 types of effector? Cool. What are the 2 types of effector? Awesome, cheese. It is, well done, cheese. It's muscle and gland. So let's get a boom for cheese. And by the way, who can tell me what the muscle does and what the gland does? So you need to know that as well, obviously. Boom. Yeah. So muscles, hopefully you got this right in the chat. Muscles contract. Oh my gosh. And glands, now glands release hormones. Glands like your pituitary gland. But there are several things in the body which act as a gland and release hormones, but they also act as an organ and produce like enzymes and things like that. Like your pancreas. Your pancreas in biology paper 1 produces amylase, protease, lipase, but it also can act as a gland and secrete specific hormones that we're going to test you on today. So, oh my days. Right, we've got a couple more questions. So we're going to do a bunch of questions on the nervous system, then we're going to move on to homeostasis and look at the control of internal conditions. So let's just do a few more of these. So what's A and what's B? Look at this nerve cell. It's got like to the brain. On the diagram, label A and B. There is a junction between the connecting neuron and the neuron carrying the impulse to the brain. What name is given to the junction? In what form is information passed across the junction? Who can smack up that? Well done. Synapse and chemical messengers. Boom, boom, boom. So well done, well done. Synapse and chemical messengers. Well done. And then we got A is going to be, look, you might look, oh, sir, man, I never seen one of these. Yeah, this is just testing you on some basics. So, yeah, anywho, watch out for that. Right, I've got a little bit of dipple, bittle, triple, whittle, slipple, triple with the nervous system. So I'm just going to cover this little bit of triple on the nervous system, and then we're going to boom off of the nervous system. I'm going to do the eye at some point, but I'm not going to do it right now. I'll do the eye later on in the live. Okay, the eye is part of the nervous system. This is a tiny bit of triple. It's only going to take 2 minutes. If you're doing triple, you need to know what this is. You need to know what this is. You need to know what this is. And you need to know what this is. So you actually need to know 4 things, but 3 here. Who can tell me what A, B, and C is? Who knows A, B, and C? It's only going to be a couple of minutes on triple here, folks. Yeah, there's going to be a little bit of triple here. Cerebral cortex, lovely jubbly, well done. So this can be called two things, cerebrum or cerebral cortex. So it has two different names. You choose. You decide. Right, that there is that. So yeah, cerebral cortex, I don't need to have extra arrows on here. The only one I need an extra arrow on is because it isn't labelled here. B is the cerebellum. Now you guys need to know that this is for balance and coordination. We've got the World Cup starting soon, and so when someone is lining up a penalty kick, they will require balance and coordination. However, your cerebrum is in charge of creativity, so where you're going to actually kick the ball, thinking, learning, language, when you're speaking, when you're thinking, when you're doing anything, all your revision is happening here in C. Cerebellum is just your balance and coordination. Now, we haven't labelled this, so I'm going to label that. Anyone know what that is? D. And then B, we've got the pituitary gland here. Yeah, okay. And above it is the hypothalamus. No, and just for those of you that are really interested, above it is the hypothalamus. That is known as the thermoregulatory center as well. And this is the medulla oblongata, but I think you might call it brainstem. And the brainstem is in charge of your heart rate. This controls heart rate and breathing rate. Unconscious things like when you're asleep tonight, your medulla oblongata, your brainstem makes sure you don't die because it keeps your heart beating and your breathing going. So which part of the brain controls balance when rolling a bicycle? Is going to be the cerebellum, which is A. And then the ears send information about sound to the brain. Which word describes the brain? The brain is a coordinator. You need to know that if you're combined as well. And then what type of cell carries impulses from the ears to the brain? What cell carries the impulse from the ear to the brain? It's going to be a neuron, a nerve cell. Now, you can say nerve cell or a neurone, okay? It's going to be the sensory neuron. If you wanted to be really specific, it's going to be the sensory. Yeah. Going to be the sensory neuron. Nice. Okay, where are we? What we doing? What's happening? I can't see. I'm blind as a bat. Right. So that was just a little bit of triple. Back to the real McGill skill. Let's do. Let's move on. Let's move on. Okay. Dodgy AI drawing. Can anyone spot the mistake? This is an AI drawing of the 6 glands that you need to know. So we just talked about glands. We're going to look at, we just looked at the nervous system, which is a way of communicating using electrical signals, mainly electrical signals that travel along neurons. We're now going to look at the endocrine system. Can anyone spot the deliberate error that AI did? Right, yeah. So we got a little bit of an error going on here. Okay, this is known as the endocrine system. It's basically your hormonal system, and your endocrine glands release hormones. And there is one in your brain that you need to know about. We just looked at that. It kind of looks like a bit of a droopy thing, and it's called the pituitary gland. Now, the pituitary gland releases chemicals. I'm going to give you just a list of some of them. ADH controls water regulation. LH and FSH, we will come on to those later. But those are just some of them. It also releases TSH, thyroid stimulating hormone. Now we have the thyroid gland. So the thyroid gland, and the thyroid gland releases thyroxine hormone. So produces thyroxine hormone. Thyroxine hormone controls, oh my gosh, does that look like control? I can't write properly. metabolism. So if that's not working properly, you might be overweight or underweight due to your thyroid gland not speeding up or decreasing your metabolism. Things like respiration, building proteins, building carbohydrates, building skin, building hair. That is your thyroid gland is controlling all of those metabolic reactions. Now the pituitary gland, actually known as the master gland, it actually stimulates the thyroid gland with a hormone called TSH, which will make it release thyroxine and speed up your metabolism. Now this is an error, so let's get rid of that. The lungs, they are an organ, so there's no problem them being in the body, but they shouldn't have these on top, okay? This was an AI error, which is good because we like to spot the error. Challenging. Really, they should be here. These glands are found on the kidneys, and these are known as, you have to know the location of these. You can just say they are found above the kidneys. They're called adrenal glands, and the adrenal glands release adrenaline. So, by the way, the arrow which I'm drawing here is like release or stimulate. So they release adrenaline. Adrenaline is a hormone, a chemical messenger, and the hormone adrenaline, it is for your fight and flight. So anytime you're going to run for your life or you're going to have a fight, so fight or flight. Oh my days. Now this is triggered when you go into an exam. If you're the type of person before you go into an exam, you start breathing fast. That's because your body thinks you're about to take on a Tyrannosaurus Rex in a fight, or you're about to fight a tiger, right? And you're either going to die or survive, or live, but you're very nervous. So when you're nervous, you'll breathe fast. You're oxygenating your muscles for respiration so you can contract them and run for your life or fight for your life. Your pupils will go wide when someone is nervous to let light in to receive as much information about their surroundings as possible. Your heart will beat faster to pump the oxygen to your muscles. Things like people get a cold sweat released. There's all sorts of things, and this is all part of the fight or flight. Some people say they feel sick when they get nervous, so you're about to have a fight. People get a sick feeling like they're going to throw up in their stomach, and that is caused because the blood is being taken away from your stomach and sent to your muscles, so you can get maximum fighting, okay, maximum oxygen in your muscles for fighting or for running. And that is all part of the fight or flight response which the adrenaline produces. Now we also have over here, come on pen, don't let me down, we also have, here is your pancreas. This is your pancreas. Your pancreas secretes two hormones you need to know about: insulin and glucagon. Now these are two hormones we'll talk about a bit later. And then we have, if you're a lady or if you're a boy, if you're a lady, you have ovaries. Now yes, ovaries make eggs, but their role in the endocrine system is to release estrogen and progesterone. Okay, and if you're a boy, boys basically have ovaries. It's just the way nature worked. They ended up down below and hanging outside the body. Why is that? Well, sperm die at 37 degrees, which is your body temp. So men could not have their gamete producing organs inside their body because the sperm die at 37 degrees. The sperm need to be cooler at like 35 degrees. So they have to be held away from the body, which is why boys' testes are outside of the body, and they make the hormone testosterone. Hence the name testes. Testosterone. Okay, cool. Just about squeezed it all on. Those are the organs, the endocrine glands. Anybody got any questions about those before I move on? Anybody got any questions, any confusion? Anyone got any questions? Yes, so what I'm talking, Mustana, I'm talking about what each of the endocrine glands secretes. I'm not talking about, like, the pancreas is an organ that also secretes enzymes. We're not interested in that. We're just interested in the chemical messaging system, which is the endocrine system. Hormones, by the way, you have to know, if you're asked what is a hormone, which could be a nice simple content one, what is a hormone? It's a chemical messenger. All right, a hormone is a chemical messenger. That's what it is. Yep. I will be going through required practicals as they come up. I'm not going to go through them right now. Adrenaline increases heart rate. That's right. Blood supply to the brain and muscles. Absolutely. You want more oxygen to the brain so you can think fast. If you're going to get in a fight, people sometimes describe in life and death situations like being in wars when bullets are firing at them, things go into kind of bullet time. And really it is just that so much blood goes to the brain and oxygen goes to the brain that the world around them can seem slower. Things can slow down in those critical moments. Not enough for you to dodge bullets like Neo in the Matrix, but still, that is all part of the fight and flight. So when you release, and if any of you ever, if you ever do get in, if you're in a life and death situation, sometimes it might only last 10 seconds, but afterwards your body is completely wrecked, drained. Like you literally, your arms are floppy and your legs have gone floppy. It's due to adrenaline, a massive surge of adrenaline. Adrenaline not only pumps oxygen to all your muscles, but it also breaks down all your glycogen into glucose. And so a minute later, you can be completely and utterly exhausted, which can happen to people if they get too worked up before a sporting event. Okay. Right. What we're going to do. All right. You guys are all good with that. So what we're going to do, we're going to go on to the control of glucose in the body. But before I do that, one thing I just want to go through, and then we're going to go on to the control of glucose, which you have to know about, and it's part of the endocrine system. So one thing we're going to deal with here, hold up. Okay, so the endocrine system uses hormones. Now, if you're asked what a hormone is, I already said that is a chemical messenger. And hormones travel around the body in the blood. So, for example, in the part of the menstrual cycle, hormones need to go from the pituitary gland in the brain to the ovaries in order to tell the ovaries to develop the eggs and release the eggs. They have to travel through the blood to get there. Hormones have to travel through the blood. Now, if you compare this to the nervous system, an electrical signal which travels very, 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 very fast. So hormones are a lot slower. That doesn't mean they're that slow, but compared to the nervous system, they're very, very slow. So hormones travel in the blood. That's one thing we need to know about hormones. Also, hormones have widespread effects. Now, what do I mean by that? Testosterone is released in boys once they start going through puberty. They start getting pubic hair under their arms. Their Adam's apple comes out, their voice deepens, all of their shoulders widen. So that's widespread effects. But if I touch this hot coffee cup and I pull my hand away, that's a very localized effect. So the nervous system has a very narrow localized effect in one region of your body. The hormones can have a big effect. If adrenaline pumps and you're scared, your hairs will stand up on your arm when you're scared. Your eyes will dilate. Your heart will beat faster. So that's a big, widespread effect. Hormones have widespread effects. And we also say that effects of hormones last longer, right? So takes a few minutes after you're scared for your breathing to go back to normal once adrenaline's released. Take a couple of minutes for you to breathe normally again. Whereas if I pull my finger away from a hot cup, done in 0.2 of a second. It's done. So you may well be asked to compare the effects of the endocrine system versus, they'll often do questions like this. So just make sure you can do that in your head. I'm not going to go through all of it for the nervous system, but just make sure you can do that in your head. Endocrine system versus the nervous system. Hormones travel in the blood. Oh, and I didn't say chemicals. They're chemicals as well. So look, 4 differences. Hormones travel in the blood. The nervous impulse travels in neurons. Hormones have widespread effect. The nervous system has a localized or narrow effect. The effects of hormones last a long time. The effects of the nervous system, the response is a short one. And then, of course, the hormones use chemicals and the nervous system uses electrical messengers. Okay, right, we're going to look specifically at blood glucose. I'll give you some questions on this in a sec, but we're going to look specifically. Now you have to notice, if you're combined, everything I've talked about here so far, other than the brain so far, has been combined and triple. So I will specify when I'm going to do like 5 minute on triple, which I'm going to do in a minute with temperature, because you don't need to know about temperature regulation if you're combined science. So what I'm looking at here is control of blood sugar or blood glucose. So I'm going to look at blood glucose regulation, and this is controlled primarily by the hormone system rather than the nervous system. So what happens? When would your blood sugar be high? At what part of the day? At what point of the day would your blood glucose level increase? That's what you've got to ask yourself. What part of the day would your blood glucose level go up? This is part of homeostasis, that's right. And I will go through what that means as well. So part of the day where your blood glucose level goes up would be after a meal. So after eating, right, breakfast, lunch or dinner, blood glucose goes high. And this happens for everybody. It doesn't matter if you're diabetic or what. This happens for absolutely everybody. After a meal, your blood glucose goes high. Now, this can be dangerous. Uncontrolled high blood glucose can lead to coma and death. That means your body can shut down and you can die. So we don't want it to stay high for long. So we have a response. So the response to high blood glucose is that certain cells in the pancreas release insulin. So I'm going to write this and then I'll do the opposite. So blood glucose high, 1. 2, insulin released from pancreas. So what you need to know, let me get rid of that arrow and just try to like make that arrow a little bit nicer. So if the blood sugar is high, insulin is released from the pancreas. What happens? The insulin puts the sugar, it puts it into the liver. So the sugar gets put into the liver. Okay. So sugar goes into the liver. Look at the word insulin. Insulin. And think of it like this. In, sugar, liver. So if you think in, sugar, liver, it puts the sugar, the glucose, into your liver. So glucose goes in here and the glucose is changed into something called glycogen. So insulin released from pancreas. 3, puts the sugar in the liver. 4, glucose turned into glycogen. The result is 5, blood sugar drops, or blood glucose drops, decreases, right? So that's one side of it. Now, everyone cool with that? Is everyone okay with that? Because then I'll bus on to what happens if it goes high. Okay. So what we're going to do next, we're going to look at the opposite, because we call this process, this is an example of negative feedback. So we're going to look at what happens when it goes low. And in fact, I might just do it on the same, let me do it on the same one. Okay, let me do it on the same thing, but let me use blue. Let's use light blue. No, let's use that. All right. Okay. Okay, so what happens is your blood glucose, what happens throughout the day, and I'll show you in a second, but your, let me do it now actually. Bish. Bosh. Okay, so what happens throughout the day is you eat a meal. In a normal healthy person, this would be like your sugar level. So let me show you this, and then I'm going to do the opposite, which you have to know. So if this is your sugar level, or let's call it glucose level, and then this is time. So throughout nobody in the world has a blood sugar level like that. No one's blood sugar level just stays constant. Yes, homeostasis means the control of internal conditions to maintain conditions at a constant level, but they're not really constant. After a meal, your glucose goes up, your insulin kicks in and brings it down. Uh-oh, it goes too low. So what happens is we go too low, but it's brought up again, down, up, down, up, down. We call this negative feedback. This control of internal conditions is known as negative feedback. So actually, that is what your sugar levels are like throughout the day. They are not in a straight line. And why is that? Because when the blood glucose drops, it's inevitably too much glucose is going to go in here, and it's going to fall slightly lower than the body wants. So the body then responds, and if your body's working well, it responds quickly, and it starts it going back up again. And the way it responds is like this. So blood glucose drops, and then we're going to go number one, glucagon released from pancreas. Now remember, that's a 2-mark situation. Glucagon, pancreas, 2-marker. And then what's going to happen? Number two, wherever I'm not going to fit this in, am I? So glucagon released from the pancreas. Number two, what does the glucagon do? You can't even see that. Glucagon reverses this, turns glycogen back to glucose, converts glycogen to glucose, and moves it back into the blood. Moves into blood, and blood sugar goes back up, and blood sugar returns to normal. Now literally, the thing with hormones is your liver is covered in cells, and they have little locks on them, and insulin has a specific shape. So when insulin binds to that lock, it literally opens the cell membrane, allowing the glucose to go in. So that's how insulin works. Hormones have target cells that they are specific to. Anywho, that is our blood sugar regulation. What we're going to do, let's just test you on a little bit of that. And then, okay, hold on. Let's not do that yet, because I ain't talked about that yet. So let's just go here and just finish off this bit on the sugar. So we got 2 types of diabetes you need to know about. So you got, and I'm just going to go over diabetes. You've got type 1. Now type 1 is generally what you're born with, generally genetic, so you're born with it, inherited, right? It's a mutation where you cannot produce insulin, so cannot make insulin. Now if you imagine here is a liver, and you want to move glucose into the liver, what does insulin do? Insulin binds like this. It binds to receptors. Here's a receptor on the liver. Insulin binds to the receptor and opens a little doorway, and the glucose can go in. If you don't have insulin, you can't open the doorway and get sugar out your blood, so you can't control your blood sugar. Very, very bad. So you cannot make insulin, so you're going to need insulin injections. So you actually need to have some insulin injected into your body, because you cannot make it because of a mutation in your genes, which means that you don't have the code to make the insulin. Now it also helps, to diet. What do I mean by diet? You need to be specific. Low sugar diet, or even better, low carbohydrate diet, and do exercise. So if you do exercise, some of this glucose is going to be used in respiration, because glucose plus oxygen turns to carbon dioxide and water. So you're going to lower that by exercising, and by having a low carbohydrate diet, you're going to have less glucose. So you can do those two yourself, but you will need injections. Now let's look at type 2. Is everyone cool with that? I hope so. Everyone cool with that. Now let's look at type 2. So let's look at type 2 diabetes. I'm going to leave that there. Also helps. That helps as well if you've got type 2. Right, type 2 is a slightly different story, guys. So type 2 diabetes, I'm just going to say it's linked to obesity. There's plenty of people who aren't obese with type 2 diabetes. So obesity. So it's linked to obesity, basically your diet. And this is different. Still make insulin. But receptors on liver are resistant. Now, what the fudge does that mean? Well, what it means is you're still making insulin. Wonderful, right? You're still making it. But the you've damaged, due to your lifestyle, you have damaged the receptors. So the receptors have changed shape. Look at the shape of this receptor. They can't bind together anymore. So are insulin injections going to work with someone with type 2? I can give them as much insulin as I want. It's like the lock and key with enzymes. They're not going to bind to the liver receptors, and so type 2 is not going to benefit from insulin injections. So they need to watch their diet and do exercise. There is a drug that can help them called metformin that they will have to allow in the exams. Just a little bit of extra. Some of you may have relatives that take this. If you've got a relative with type 2 diabetes, then they will take metformin. Okay, but insulin injections will not work. So insulin injections do not work because the person still makes insulin. Their body just doesn't listen to it. Insulin, or has become resistant to it. Insulin injections do not help. Okay, cool. Now we're done that, let's bus on and get a move on. Let's see if you can do this question. So you just said what causes someone to develop type 2 diabetes. Now there's a number of factors. A lot are inherited genetically, some are genetically inherited, but your lifestyle, so being overweight, having a diet that's high in sugar, if you're just always eating chocolate and sugar and crisps, high carbohydrates, if you love your white rice, not good. If you love your pasta, your noodles, not good. And we see quite a lot of areas of the world which have become more wealthy, and as they've become more wealthy, what's happened? The amount of diabetes has gone up. It used to be like in the UK and in the West that diabetes was very, very high, type 2 diabetes, and in the East it wasn't so high. But now what we've found, like just looking at the data from China and places, like as they've become the richest country in the world, guess what? Their type 2 diabetes has gone through the roof, and it's linked with obesity. So they've got a huge obesity problem, just like that we have in America and Europe, and they have their diabetes levels now match ours. So it's to do with your diet, your type 2 diabetes. Diabetes type 1, it's your lifestyle and your diet. And you might think, oh, well, I'm reasonably fit, but look, if you eat lots of sugar, you shouldn't really need to add sugar to anything. So, and too much carbohydrates, too much rice, too much sugar, too much pasta, too much noodles, basically explains it. That's what leads to type 2 diabetes. Low carb diet and exercise. Yes, you can have a boom. Boom. Okay, so name the organ. This organ, some of you might have put pancreas. This is the liver that this one is pointing to. The pancreas is actually here. That's the pancreas. This is the liver. Organ A stores glucose. Name the hormone. Type 1 diabetes have to inject. That's going to be insulin. So if you have type 1, you've got to inject that. Why should a person with diabetes not drink too much sport drink? Because it is high in glucose or sugar, high in carbohydrates. Other than using drugs, give 2 methods of treating diabetes. So this is any diabetes, will help with exercise. And now don't just say diet, because a diet can be, I'm on a high sugar diet. So the examiner might say, this kid thinks that if you have a high sugar diet, it treats diabetes. So say low carbohydrate diet or low sugar diet. All right, awesome. Carb controlled diet. Yes, 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 yes. Now, what are we doing? Sir, do you have type 2 fat boy? Oh, hell. Now, what, Ishal, I hate you, bald boy, Petrovic. Oh, I got something for Petrovic. Don't worry. You know, we got one of them in the chat. You know, when you get these idiots in the chat, you got some real plums. Guys, guess who that was in the chat? Have a little guess. Have a little guess who that was. Yeah, you know that dude. That was Petrovic. What can we say about him? He's got issues. So we're going to time out his backside. Give him a 10-minute. There you go. Petrovic is gone, guys. Oh, what can I say? Occasionally you get some bell pieces in the chat, and there's not a lot you can do about it. It's just part of life. So, you know, what we got to say to people like Petro, get the beep out of here. You have been destroyed. Okie dokie, okie dokie. Back to business, back to business. Let's go on. Right, this next bit is just a couple of minutes. 2 minutes a triple. Ugh. Not liking this image. So 2 minutes a triple here, guys. So just hitting the trips for 2 minutes. Yeah, what do we have? What do we have? What do we have? So we have, I know it looks like an ingrowing hair. Yucky, yucky. You do need to know about temperature response. So let me just shrink this down a little bit and then go through a little bit of the thermoregulatory response. So what you have to know if you're doing triple, and this is going to be about 5 minutes of triple on this, and then we're going to move on and look at some other stuff before we wrap up this bit of it. So look, here's the old brain. This is supposed to be a brain, right? In a head. So you have an area of your brain called your oh my gosh, regulatory center. You can call this the hypothalamus. It's up to you, right? That thermo just looks absolutely foul. So I might have to just ace that. Give me 2 secs. Oh man, what is going on there? All right. So we've got the thermoregulatory center in the brain, and the thermoregulatory center is got receptors. So it has temperature receptors, and this is where you've got a link, if you're triple, a bit of the nervous system here going on. So if it detects like a decrease in temp, what does it do? It sends an electrical signal to the skin. All right, so it's going to send an impulse, and we should probably tell the examiner we know that that is electrical. So it's going to send an electrical impulse to the skin, and then we have to know, so this is if the temperature goes down. So you need to know if temp goes down, how are we going to respond? We're going to make hairs stand up. Why? That creates an insulating layer that will insulate your body. It's going to cause these blood vessels to constrict. You're going to get vasoconstriction. Now, you can, if you don't remember that term, you can just say blood vessels narrow their lumen. But guess what? It's easier to say vasoconstriction. And this means, what does that do? Less heat lost by radiation. So you radiate heat from your blood constantly to the surroundings. Look at my red face. So I'm radiating heat to the surroundings. Vasoconstriction, you're going to go pale white because your body is trying to retain the heat. So it's going to constrict your blood vessels. And another thing, it's going to reduce sweating. How does it do that? It sends a message to these holes, these sweat glands, and less sweat will come out. So reduce sweating, less heat lost. How does heat get lost through sweating? It's by evaporation. Now, And there's also a fourth one that you might have thought of, and that is shivering. Now I've run out of room, but shivering, your muscles contract and relax rapidly, which is aerobic respiration, which releases energy, which creates heat or transfers heat to your body to warm you up. So the opposite of this is your hairs lie flat, vasodilation, the blood vessels widen to allow more. So if your temperature goes up, we say the thermoregulatory center detects this, receptors. They send an electrical signal to the skin, and then the following happens. Your hairs lie flat. You get vasodilation. The blood vessels widen to allow more heat to be lost by radiation at the skin surface. You get an increase in sweating from the sweat glands, so more heat is lost by evaporation, and obviously you won't shiver if you're hot and you're trying to cool down. So anywho, that was just going through that. Make sure you know both of those. Let's have a little look. Yes, Violet, shivering contracts your muscles and releases heat. All right, have a go at this one. What's the correct answer here, guys? Then we'll move on from this. Oh my gosh, I can't even see what that says. When the core body temperature begins to fall, changes happen to the body. Which two changes will happen when the core body temperature begins to fall? So when your body temperature, we don't want more blood through the skin capillaries. We want less blood because our body temperature has begun to fall and decrease. We want to warm up. So we're going to go for muscle shivering, because shivering releases heat. What from? Via respiration. Anytime you contract your muscles, you need the respiration to contract the muscles. And then if our body temperature has reduced, we don't want to make more sweat. Blood vessels supply the skin constrict. That's what we want, and that is called vasoconstriction. But you can just say blood vessels constrict. It's not actually the capillaries that constrict. That's why I prefer vasodilation and vasoconstriction, because technically capillaries can't change their lumen size. So notice they say the blood vessels supplying the skin's capillaries. So just in this one, thermoregulation, just use blood vessels. Do not try to get fancy and say capillaries, because you'll mess up and lose a mark. All right. Let's go to some combined business. So we're going to have a little look at the menstrual cycle. I know there's the kidney and the eye. I'll come on to that later. I just want to go through a bit of the menstrual cycle. Let's do the menstrual. Right, you have to know the menstrual cycle, whether you're combined or whether you are whatever. So let me do a quick drawing here, guys. I can't even ask my AI to do me a drawing of the female reproductive system. It won't allow it. It's like, oh, this is restricted content. So it doesn't let me. And that's why you're going to get my... Remember, GCSE, my GCSE art teacher was a lunatic. He somehow thought I was only level 4. I think he was ill. Look at this art. Look at this. Look at this. Incredible. That is the worst female reproductive system I have ever seen. Diagram of one. And here we go. That's the female reproductive system, apparently. Now we're going to link that to the 28-day menstrual cycle. So we have day 1 to 5. Then we have day 5. Let's go to 14. And then we're going to zing back round to day 28 and then back to 21. So you get the coup, right? Now I'm going to go through it, and there's 4 hormones, and we're going to go over how they control the menstrual cycle. So pay attention. Right. We need to science this up. So we're still in the first unit. Got a little bit more to do to get through the first unit, but there's only we're still in the first third, I should say, because there's 3 parts and we're still in the first third. So what am I going to say? What are you going to do? Yeah. Oh yeah, don't forget to hit the like, folks.
If you push the button,
Like me.
Would you like it?
Like.
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 do it. Do it.
Do it.
Yeah, push the like, folks. Push the like. Push the like. It helps the old. It helps with the algorithm. Okay, what we doing? Let's get on, because the more I waste, the more the dilly-dally, billy-bally. Right, day 1 to 5 of the menstrual cycle. So the menstrual cycle is a 28-day cycle. It doesn't follow like January the 1st to January the 28th. Every woman on the planet's menstrual cycle, well, not every woman, but starts on different days. Anyway, enough of that. Day 1 to 5, what happens? Uterus lining shed. Do we need to know that? Not necessarily, but the uterus lining is shed. That's the bleeding period, roughly 5 days. Day 1 to 5. And we have this, the main thing you've got to know, FSH is released from here. FSH is not released from there. I tell a lie. Tell a lie. Ask me no questions, I'll tell you no lies. Okay, so let's have the brain up here. Oh, come on, T. That is supposed to be a brain. I know what you're thinking. How can one man draw so good? I can't tell you. It's a gift from God. So here's the brain. And FSH is released from the brain. It's a pituitary hormone. So in the pituitary gland. And what does FSH do? FSH ripens. I could have done a better word than that. Develops the egg, matures the egg. So ripens the egg. Now, another name for egg is ovum, so don't get yourself confused. Ripens the egg. But it also has a second job. So those of you that can cope with 2 things, if you can only cope with 1 thing, just know that FSH ripens the egg and you'll probably get away with full marks. But if you have to know 2 things, FSH stimulates estrogen release. So FSH, right, ripens an egg, causes an, well, it doesn't do that. It doesn't cause an egg to be released. God, he's having a nightmare, isn't he? It ripens an egg. So we get a little egg going, woo! FSH ripens that egg. But it also stimulates the ovary. And this, peeps, is the ovary. It stimulates the ovary to release one of the 2 ovarian hormones. And that is estrogen. So once FSH does that, estrogen steps up to the plate. Now, estrogen is operating, so FSH is day 1 to 5, and then we're hit with the estrogen, which happens between day 5 and 14. So estrogen is released. What does estrogen do? This is a key word here. Estrogen develops lining of uterus. What does that mean? Well, let me show you. Come on, pen. Oh my good God. Sometimes this doesn't want to know me. Wee. So, develops the lining. So this is what the estrogen does. The estrogen, remember, the lining has just come out during a woman's period. They've had 5 days of bleeding. They've lost their uterus lining. So estrogen is going to build that lining back up. That's estrogen's job. However, I'm running out of room. Estrogen also, here's the key bit, which I don't have room to write because I'm an idiot. I don't think about these things before I do them. Estrogen inhibits FSH. Listen, if we've got to the point where we've got an egg ready, right? An egg is ready. FSH did that. FSH ripened the egg, got it ready. Estrogen, FSH then says, go estrogen, your go. And it stimulates estrogen's release. So FSH doesn't just ripen the egg, it tells estrogen, it's like a relay race. It hands the baton on and says, your turn. Estrogen builds the lining, but estrogen tells FSH, go away now. Don't ripen any more eggs. We've got 1 here that's ready. We don't need you to ripen any more. Don't waste any more. So estrogen tells it inhibits FSH. Guess what estrogen does? It stimulates the next hormone in the chain, and the next hormone in the chain is LH. So estrogen stimulates LH. LH is known as luteinizing hormone, and this releases egg on day 14. Now, we call this ovulation, and ovulation happens around here, so this is where LH gets busy, and that, by the way, folks, is called ovulation. I don't really have space to write that out. Guess what LH does? LH inhibits estrogen and stimulates the release of the next one called progesterone, which also feeds back and inhibits LH. So what I would suggest you do is you learn the order. It goes FSH, estrogen, LH, progesterone. Each one that comes about stimulates the next one, and the new one tells the old one to go away, and we call that inhibiting. So each one stimulates the next, and then the new one inhibits the old one, and it keeps it all under control. We call this negative feedback. It is an example of negative feedback, so it doesn't let it get out of control. Now, progesterone, what does progesterone do? Progesterone maintains the lining of the uterus. Progesterone will only be high when you're pregnant. So if you have a baby growing here, you don't want to have your period, because what will happen if you have your period? Your lining will come out and the baby could come with it. So we don't want that. You may have minimal light periods. If you have heavy periods, you have to see the doctor, because obviously there's a risk of miscarriage there. So progesterone maintains the lining of your uterus throughout the pregnancy. If you're not pregnant, your progesterone levels drop and the cycle goes back to day 1. Everyone clear with that? I'll run through one more time just in case you didn't remember. FSH ripens the egg. The egg and FSH does 2 jobs. It stimulates estrogen release. Estrogen thickens the lining of the uterus, or develops it, and it inhibits FSH, and estrogen stimulates LH. So 3 things for estrogen. Then LH releases the egg, inhibits estrogen, and stimulates progesterone. Progesterone inhibits LH and maintains the lining of the uterus. So how does this apply? By the way, FSH and LH are made in the brain, and estrogen and progesterone are known as ovarian hormones. They're both made by the ovaries and produced from the ovaries, is what we have to know for GCSE. Now, couple of things. If you're having trouble getting pregnant, so if we have a lady, and this is the most rubbish drawing. Okay, here's my lady. Okay, that is not... Oh, man. I should have got a level 5 minimums. What do you reckon? Level 5 minimums. Right, what we do is we give this lady, if she's going to have IVF, which means in vitro fertilization, we're going to give her LH and FSH. Why? Well, FSH is going to mature, develop, ripen, whatever you like to say, eggs, and LH is going to release eggs. So perfect treatment for someone who's having trouble getting pregnant is to inject them with LH and FSH. However, let's say she doesn't want to get pregnant. If she doesn't want to be pregnant, what do we want to give her? Oh my good gosh. Contraception. We're going to give her estrogen. You might say, but won't she go around with a really thick lining of her uterus? Yes, she will do. But remember what estrogen does and progesterone do. Remember what these two do? They inhibit LH and FSH. So if these are high, you can't get pregnant because you can't release eggs or make mature eggs. So they inhibit FSH and LH, estrogen and progesterone. So they put them in the pill, the contraceptive pill. Okay, cool. There are some other contraceptions you have to go on know about. I'm not going to go through all of them. I'm going to give you some questions in a second, but you need to know about barrier methods. Barrier methods create a barrier so sperm cannot meet egg. Remember, to get pregnant, sperm must meet egg. You can't get pregnant if you put a barrier in between. That can be the female condom, the male condom, or any other thing they talk about. But you can also take things that kill sperm. There's something called an uterine, intrauterine device, and that is like a copper wire the woman can place in the vagina at the cervix. So you can place about here, and right about here, and this can kill sperm. Okay, so there's things that you can put there. You can use spermicides, chemicals that can kill sperm as well. That is chemical contraception. You can always just not have sex, then you probably won't get pregnant. And in really drastic methods, you can be sterilized, which in a man means the tubes to your testes are cut. So your testes make sperm in a man. So if you cut the tubes, the sperm can't get out. They can't like go out the penis. Or you can tie the oviduct, or you can even tie the sperm ducts, the tubes that carry the sperm, but they could untie, and so you could still get someone pregnant. If you wanted it permanent, you would cut the tubes, and you cut the oviducts if you wanted to permanently not have kids. Anyway, let's get on, do a couple of questions, and then move on. So, describe how the hormones FSH, estrogen are involved in the control of the menstrual cycle. You just need to talk about FSH, estrogen, and LH, not progesterone. Worth 3 marks. Description, just say what each one does. Boom. Oh my gosh, we've got another bell end in the chat, the old footy news. What a bell piece, what can I say? You know, some people just get excited, don't they? They sit there in their underpants watching, they're looking for any live to sit there and get excited about because they've got sad little lives.
What can I say next?
Guys, all right, you get some losers. Sometimes you just got to ban their man. So there you go. Describe how the hormones FSH, estrogen, and LH are involved. So we just want to say like FSH matures eggs, and it's a description. So we can probably just say this. Estrogen, you'll probably get all the marks from this. Estrogen develops uterus lining. And then we want to say, oh, Sugar Ray Leonard. You want to say LH releases eggs. Now, if you can use the word ovum if you want to be fancy, ovums. Now, if you want to get really fancy, you could say FSH stimulates estrogen, and estrogen inhibits FSH. Okay, especially if they said the word control, so maybe they want that, and then the estrogen stimulates the release of LH. You see, each one stimulates the next one, and the next one inhibits that one, and then you could sort of say that if you want, LH inhibits that. So, but that would be like extras. Personally, I think it's enough just to say what they do. Now have a look at this one. What are glands A and B? And how does FSH move from gland A to B? Then we're going to move off of the menstrual cycle, and we're going to look at a couple of the other things, maybe the ruler drop test next, and then we will bust out of this topic maybe. I've got to do plant hormones as well for the trip people. Oh my God, it's hot. I'm flaming hot. Right. Sorry, listen, let me give you a word of advice. If you're watching and there's some people in the chat that are annoying you, don't bother responding to them or getting drawn in. Just click on the 3 dots next to their name in the chat, and then just put like remove all messages, and then they can go ahead and say whatever they like to you, but you can't watch, and then you can concentrate on getting a good grade. Okay, so that's the way to do it. Yeah, so what we got going down here? What we got? Oh, Petrovic is back. What a bell piece. Yeah, well, I'll get rid of him in a minute, but you know, until then, let him. He's probably got a semi on, so let him just like sit there and get excited. You know, it's probably the only thing going well for him in his life. If I kick him out for too long, he'd probably yeah, bad things could happen. So, name glands A and B. A is the pituitary gland, and B is the ovaries. And then FSH controls changes in gland B. How does FSH move from gland A? So how does FSH get from there to there? So this is just testing you how the hormones move. So in, blood, bish, bash, bosh. Right, let's go do, we're going to do the ruler drop test in a second. Let's just give you another one there. See if you can do this one. And then I'm going to have a coffee break when I get to the end of this. The menstrual cycle is, and the ruler drop test will be, but I've got to do the plants. So, I've got to do plants as well. So, who's got this right? Who's got this right? FSH and LH. Yes, FSH and LH. So, the woman is having IVF. What would she give her before IVF? We're going to give her FSH and LH. Mature the eggs, release the eggs. Right, we're going to have a look at the ruler drop test. Give me 2 seconds, please, people. 2 seconds. Here we go. This is a core practical for combined and triple. All right. It's a great practical. Absolutely incredible. Probably the best practical. Right, ruler drop core practical. So what do we need to know? For a start, when you talk about the ruler drop practical, to make your writing easier, we call the person who is dropping the ruler, call the person who's dropping the ruler the dropper. Just makes it dead easy. Instead of going, yeah, the person who is holding the ruler, just say the dropper and refer to the other person as the catcher. Okay, so we're just going to refer to the other person as the catcher. And that way it's much easier to talk about your ruler drop test if you talk about a dropper and a catcher. Now, what do we need to do? Well, couple of things. I'm going to go through it with you. It's mainly about the controls here. So you're going to test someone's reaction time. The closer they are to 0 centimeters, this is going to be fast reactions. This is someone who has slow reactions. They're going to catch it higher up. So fast reactions, slow reactions. We might do a test where we give them Red Bull or coffee. Why? Because it contains the drug caffeine. So remember, we'll often talk about the drug caffeine, and we want to find out if it makes people faster or slower. But we might not be doing caffeine. We might be doing left hand versus right hand. Do people have better reactions with left or right hand? I'm going to want to do 10 people with a left hand, 10 people with a right hand. I want to make sure none of them have had caffeine. I want to control their age, their diet, their weight, all of that kind of thing. And I definitely want to control when the dropper drops it. So the dropper must drop it randomly. So let me write the key things that are generally marked. Dropper must drop ruler randomly. So you're going to want to drop the ruler randomly. Don't let the catcher know. Then, where does the ruler start? Where do you start with the ruler? Start with 0 centimeters between, or actually, let's not say between, let's say above catcher's hand. So what am I saying by that? If this is the catcher and this is their hand, they got 12 fingers or something with my drawing. You want to start where 0 centimeters is there every time. That's where the ruler should start. So they just got to do that. Right? And we want to explain that. Catcher, rest arm on table. Why? Same height every time. So things like that. Rest arm on table. I'm getting messy because I'm trying to get through quick because I know people are like, so I want to get off this topic. Okay, so dropper, same diet. Why do I want to have same diet? I mean same diet on the day. Like they didn't have extra coffee. If you're doing coffee, they didn't have coffee, 12 coffees before the test and that kind of thing. So that's the kind of thing. And then of course you want to repeat, remove anomalies. So, You get the idea. Any questions about this, just call them the dropper and the catcher. Make sure the finger and the thumb are the same distance apart. Make sure they rest their arm on the table. Make sure 0 is there. Make sure they randomly drop it. The dropper randomly drops it and they can't read their body language. Why might they get better? Why is this experiment a bit rubbish? Because as they get better with practice, their reaction times might get better just because they're guessing when the drop is going to happen. So all of that kind of stuff might happen. This is a nice test. You want to have 10, same gender, same age, because age and gender might affect reactions, same things like that, and what they've had to eat, like if it's an energy drink, a Red Bull or a Monster, it will contain caffeine. So that kind of stuff, keep all that the same, and that's your ruler drop test. Right, I think I'm going to go through plant hormones, which is triple. So I haven't done mRNA yet. Yeah, oh, something else really important, very important to note, is if you do this with a computer, and you can go now onto Google and Google reaction time test, and on Google will come up a reaction time test, and it will go red or green, and it will say like click as soon as you see the screen go green. So it'll be red, and then it will go green, and you click a button, any button. Now that is far more accurate because the timer, it's a computer's timing, is going to be more accurate, but also there's less bias, B-I-A-S, because you can't guess when the computer is going to change color. It will randomly change color, whereas you can work out when your mate's going to drop it just by the twitch of their shoulder or something. So you might be able to read their body language. So there is bias in having a person do the reaction times. Okay, last little bit on this, just a couple of questions, just while I grab a water, see if you can do these, and then we're going on to, plant hormones, which is only for triple. We'll be on that for like 5 to 10 minutes, and then that is the end of this unit, and then we will go on to genetics, variation, Punnett squares, all of that business, selective breeding, natural selection, speciation, classification, everything around the nation, whatever. Yeah, you get the idea. Fatty Petrovic sitting in his underpants getting excited again. Petrovic, what a loser. Boy, put the pills down, boy. Go and do something, yeah? Go and do something interesting with your life. Or maybe you shouldn't. Maybe you do us all a favour. What a loser. What a sado. I'm sure there's another. I'm sure. Sir, go prison. No, you mean go to prison. Oh my gosh, this geezer, man. Yeah, don't worry. Don't worry if you're here for the A-level lives. Petrovic won't be, because I don't think he's making it that far. But not that that matters, because there will be some. There are plenty of toilets in Aberdeen that need cleaning, so he will always have employment. And there's other jobs Petrovic can look forward to. So I'm sure actually toilet cleaning takes a little bit of skill. You've got to handle dangerous chemicals like bleach and stuff like that. And that might be a bit dangerous. They probably can't let Petrovic alone with the bleach. Do you know what I mean? Because it'll all go a bit pear-shaped for him. So, you know, like, what can I say? Sado. Right. Back to the live, because I was going to go get myself a drink. Anyway, I'm only hammering people who are saying rude things. You know, I'm not going to hammer you for just having a laugh, but if you're getting a bit too excited, you've got to be dealt with. Which gland is the pituitary gland? So the pituitary gland, if you were on this, is A. Which gland is the pancreas? That is D. Which of the following is a target organ for insulin? Oh, a target organ for insulin. Well, that's going to be the liver. The liver locks on to the insulin locks onto the liver and lets the glucose in. The glucose is stored as an insoluble substance. What is the insoluble storage substance that is formed from glucose? So we never want to keep glucose as glucose, because it has an osmotic effect. So we convert it into glycogen, and there we go. Bish bash bosh. Right. Oh, we've already done that. I already did that. That was a 3 marker, and this is a 5 marker. We're not doing that again. Right, we're going to go on to plants, guys. So I'm going to grab a drink and be back in 2 seconds. Look, actually, while I go grab my drink, I need to leave you with something to do so you don't get overly bored. So, yeah, see if you can do that. I'll be back in 2 shakes. Okay, here we go. So, name the structures labeled A, B, and C. So, oh my gosh, difficult this one. It's hard to see. You can see here, this is a stretch receptor, and the receptor, if we follow it round, is connected to A. Which neuron is connected to the receptors? That A is the sensory neuron. Don't beat yourself up if you got this wrong. This diagram's quite difficult. It goes up here to C. C is actually part of the spinal cord. So that was a bit of a beast. So spinal cord or spine, they'll probably give us the mark for spine. And then over here, B is the one that comes down to the muscle. And look, it kind of branches into the muscle because it's going to spread its electrical signal through your muscle and make you contract it. So B is going to be, oh, hold on a minute. What a twit. Got those two wrong, didn't I? Yeah, B is the motor neuron. I did them in order. So this one here, B, is the motor neuron. And yeah, let's go spine there. I mean, spinal cord, really. What is the effector in this response? Stretch receptor. You can see that is a muscle. It even labels it for us. So the answer is muscle. Right. You can say CNS, central nervous system. Yeah, you can do. Okay. No, I live off coffee and coffee is not good for me. Right, guys, this bit is for triple only. It's going to take about 5-10 minutes. I say that. That normally means another 50% on top, so 15 minutes. But yeah, it should take 5-10 minutes, but I have a verbal diarrhea. Right, what we have here is plant hormones. And this is known as phototropism. So I'm just going to quickly smash through this for you. So what happens is light hits the shoot of a plant. I know it's hitting the leaf, but I'm supposed to be focusing over here. Light is hitting here, and auxins are hormones that move away from light. Okay, auxins do not like to be in the light, so they move to the shaded side of the plant. So I'm going to write what happens here, and this is phototropism in, this is really important and this is quite difficult, but phototropism in shoot. So what we want to say is, now even if you flop the overall bit of this, there are certain bits that you can always pick up marks. So auxins, they are growth hormones, but auxins move away from light. Okay. Cause the dark cells or shaded cells, the cells away from the light, cause shaded cells to elongate. Now there's several ways we can word this. We can say uneven distribution of auxins means uneven growth. So they cause these cells in this half, imagine this is the dark half and this is the light half. The dark half is growing more. It causes the shoot to bend because one side is growing more than the other. So causes the shaded cells to elongate, grow more. You can just say to grow more. And then we can just say, if we want, you could say uneven growth, bend towards light. Okay, that is known as phototropism. It's a light response. So plants respond to light by growing towards the light. How do they do that? The auxins, the growth hormones, move to the dark side, make the dark side grow more. That uneven distribution of hormones or auxins causes an uneven growth pattern where the dark side grows more than the light and it bends towards the light. So that is phototropism. And yeah, auxins are, oh my gosh, growth hormones. But it gets a little bit more tricky, so let's go. Look at these roots. I mean, they're never going to ask you this, but yeah, they're never going to ask you that, so don't worry. Right, let me move on. Oh my gosh, what happened there? We had a little technological explosion. Right, I'm back. We're back. Okay, so watch out, watch out, watch out, watch out, watch out. If you get a phototropism, check the writing. This is super important. AQA are sneaky. They will tell you that the seed, they might tell you a plant is in a dark cupboard or the plant is underground. Guess what? If it's underground or if it's in a dark cupboard, it cannot be phototropism. So RTFQ, read the question. Because I have seen it where kids start going, yeah, the auxins move away from the light, and yet the question said it was in a dark cupboard. So immediately they say it's in a dark cupboard. You know you're dealing with gravitropism. This is also known as geotropism. So it's geo meaning earth, towards the earth. It's up to you, with gravity or towards the earth. You can call it either one. They're both scientifically correct. Now, this is where it gets difficult. Okay, in root cells, it's going to get real difficult here. In root cells, auxins, they do the opposite. Auxins inhibit growth, right? They do not elongate. They do not increase growth like they do in shoot cells. This here is called the root. This here is the shoot. They have a different response in both. Let me go through the response first in the root, then the shoot. Then we're kind of nearly done. So you want like 4 bullet points to remember. It's difficult to remember, but we're going to say gravity. This time it's not light. So you're going to get marks from saying gravity pulls, even if you get the rest wrong, gravity pulls auxins to the bottom of root. So gravity pulls auxins to the bottom of the root. Then we're going to say, not elongate, inhibits growth. Let me rub that out. So that's very, very difficult, but auxins have a different effect in the root and the shoot. So let me show you what's happening here. The auxins in here, in the root, have gathered at the bottom. They cause the top. This time the auxins are at the bottom, but this time they stop it growing. So the top grows more and it bends down. How do we say that? Well, we say auxins pulled to the bottom of the root by gravity. So I've said that gravity pulls auxins to the bottom. They inhibit growth, uneven growth, root bends down. Now, I want to run through the shoot bit because there's a shoot bit here. That, by the way, is called, if you want positive and negative and you got that on your syllabus, then that is known as positive gravitropism because it goes with gravity. Now, let me give you the shoot. The shoot underground is different, right? Look, you're a seed underground. How the beep does a seed need to go? No. Imagine a seed underground. Its shoot came out and it grew down. It's going to die. It's never going to hit light, never do photosynthesis. So how can this seed know? How does it know to go up? Is it intelligent? Does it have a nervous system? No. Plants do not have a nervous system, no electrical signals. What they're doing here, what they're doing here is using a hormone system to sense their environment and to send the shoot up and the roots down. How does this work? So it works slightly differently. So what happens here? This is a shoot, not a root. And auxins in the shoot make cells grow more. We're going to do the same thing, but we're going to say auxins pulled down by gravity. So gravity pulls the auxins down to the bottom part of the shoot. We're then going to say auxins elongate cells or cause growth in shoot cells. Elongate shoot cells. So they cause more growth. This time the bottom is growing more. I'm going to bend it upwards. So auxins elongate shoot cells. And then I just want to use like things that I know are on the mark scheme. Uneven growth. Maybe uneven distribution of hormones is good as well. Uneven distribution of auxins. Uneven growth. Shoot bends up. Now, you know it's going to bend up because it has to break through and then start photosynthesizing. It's got to get through the soil, but the roots have to go down. So the whole thing just changes in that auxins in the shoot cause growth, auxins in the root inhibit growth and make it bend down. Now that's negative geotropism because it's growing up away from gravity, whereas the roots going down is positive geotropism. So anyway, that's all the tropism, smopism, hopisms. There's also hydrotropism if you grow towards water. And then a couple of last things to know about hormones, and then I'm done with this unit for now. I might come back to the eye later in the kidney if I have time, but they're triple topics that take a little bit more time, so I might come back to them later. Gibberellins. Gibberellins, you need to know, do three things. What do? They cause seeds to start growing. We call that germination. What else do? They cause fruit development. They make fruits larger. So as a farmer, if I get the gibberellins out and put it on my crops, the fruit's going to get bigger apples, bigger oranges, etc. And they help with plant sexual reproduction by making flowering, encouraging flowering. They start flowering, so they cause your plant to get big flowers. So you need to know that about gibberellins. And the last thing on the hormones, this is a picture of ethene. Bananas give out ethene. So ethene is a chemical and it's released by certain plants. And when it is released, bananas give out a lot of it. It causes the fruit around it to ripen. So don't put your bananas with the rest of your fruit, because if you put the bananas with the rest of your fruit, all of your fruit will go off quickly because it will speed up their process of ripening. So ethene causes ripening. Now, where does most of the fruit come from in the UK? We have to ship it to England because we don't have the kind of weather where we can grow tons of fruit. Yes, we can grow apples and stuff, but you can't grow mangoes and God knows what else here. You need too much sunlight for that. So what we do is we ship it here while it's green and not ripe, not ready to eat. And then we take it to Tesco and Waitrose and Sainsbury's. And then what happens is they spray it with ethene to make it ripe. They pick it while it's still unripe. Okay, and it would normally stay on the tree of the plant longer, and then ethene would be released by the plant and it would ripen at the end in the last few days. We pick it while it's green, and that way when it's transported it's harder, so if it falls, a big crate gets knocked over on a ship or a plane, they don't all squish, they're harder. And then when it gets here and it's going in the trucks to the supermarket, they can spray ethene in there and trigger it to ripen. So that's just a use of one of the ways that we can use ethene. Plants naturally produce ethene in a few days, just in the last few days when they want to ripen it. Now bananas happen to produce tons of ethene, so keep them away from your other fruit. Okay, we're going to go on to a different topic. Anybody got any questions about that topic before we move on? Which practical? I kind of loosely did the ruler drop practical. I'll find some questions later. I'm going to go over, let me just zing. I think we're going to start the ecology topic now. I might start the eco topic. I'm going to do the eye later maybe. I've got questions on the eye lined up, but like the eye is only for the trips. So was the plants, to be fair, but let me go on to the old... Okay, let me just see. Let me just see what you gotta know. Plant hormones. Done, dusted. Okay, did I talk? Was cuttings and that there? No, cuttings isn't there. Okay, I didn't talk about the thyroid gland. Thyroid gland controls your metabolism, and your pituitary gland, called the master gland, stimulates the thyroid gland. Like if you've got a low metabolism and you need to speed up respiration, the pituitary gland will send a signal, a chemical message called thyroid stimulating hormone, so in the blood, down to the thyroid gland, and the thyroid gland will release thyroxine, and thyroxine will speed your metabolism up, make your chemical reactions happen faster in your body. Now if they're going too fast, then the pituitary gland will not release TSH, the thyroid will not be stimulated, and it will not release thyroxine. There we go. Right, I'm going to go through DNA, the genome, mRNA, a little bit on protein synthesis, mutations, reproduction, and meiosis in the next bit. So that's what I'm going to go through now. So I'm going to quickly smash through that. So here we go. What we doing? Okay, pituitary gland is the master gland. It's just a nickname for it, so yeah, best to call it pituitary gland. Right, let's look at DNA. Okay, so let me just do this. Oh my good gosh. So if you look at a cell inside the nucleus, we got 46 chromosomes. This is a human cell, 46 chromosomes. So this is a chromosome. If we get inside a chromosome, you're going to find a long piece of material that we call DNA. Now, you have to know this if you're combined or tripled. That's the crustiest bit of DNA ever. Deoxyribonucleic acid. You do not need, you don't need to know how to say that. Chromosomes, DNA inside the chromosome. Now, what is the shape or the structure? It has a double helix structure. We call it a double helix. That may come up as in a 1 marker. So it has a double helix structure. Now, I will branch off to just show you a tiny bit of triple here, and that is that this is a polymer, 2 polymers. So it is made of 2 polymers. You know, if you're in chemistry, a polymer is a long chain. So it's made of 2 polymers. However, what are the monomers? All polymers are made of monomers. For instance, proteins. People get protein and DNA mixed up all the time. Proteins are made of amino acids, and DNA is made of nucleotides. So the monomers are called nucleotides. So a single monomer of DNA is known as a nucleotide. DNA makes protein, codes for it. It isn't protein. Nothing to do with protein other than that it's the instruction for protein. It'd be like confusing the recipe for a meal, saying the recipe is the food. The recipe is the code for making the food. You wouldn't say, oh, this is a lovely recipe. You know, you get the idea. Anyway, enough of my whiffle waffle. Monomers are called nucleotides. So that's why it's deoxyribonucleic acid, because it's a big long chain of nucleotides. What are they made of? So they're made like this. This is a phosphate. You have to know that is a phosphate. It's attached to a sugar. It's actually called ribose, a ribose sugar, but I'm just going to give it an S. And then it has something called a base. So that is a nucleotide, a phosphate, a sugar, and a base. Okay. Now, for those of you, that is a single monomer. Now, how do they all connect? Well, I'm just going to show you it connecting over here. So, bish, bash, bosh, sugar. And then what happens is we just draw another one there. The phosphates and the sugar are identical. Every nucleotide has the same phosphate and the same sugar. And we actually call this the sugar phosphate backbone, meaning that it's like the skeleton, it's the same. Where DNA is different is here on these. And these can be A, T, G, or C. So I could have done another. Let's do another. All right, let's smash that up there. Oh my gosh, that's ugly. No, that's connected to 2. What the arse am I doing there? That should be connected to the bottom of that. Whoops. Whoopsie. So that would be connected there, and then that would be connected to another one there. Okay, so I just wanted to get them all in. So C, A, T, G, C. Now that is just one of these. So like if I pull, what the arse? If I pull those apart and then put them together, That is just one of those strips. That's one of the polymers. There's another strip, although they're twisted in this double helix, called an alpha coil. Now, you do have to know that they join up in a special code. So C, always H bonds, hydrogen bonds, to G. Cecil and Gary, and Terry and Ari. Yeah, anyway, they go like that. A and T, G and C, like that. That's how they code. You might get tested on that if you're in triples. Right, going back here, we have, you may be asked, what is a gene? So you might be asked, what is a gene? Now, a gene is a small section on one of these strips. So it could be this bit here, the gene could be there, or the gene could be there. So what is a gene? A gene is a short section of code. A gene is a short section. By the way, when we say short section, it could have 10,000 of these bases. Each one of these is called a base, by the way. So that is phosphate, sugar, and a nitrogen base. Now, each one could have 10,000 bases. So when I say a short section, your DNA is pretty long, right? So it is a short section of DNA code, or DNA, and then we could say that codes. This is the long answer. I'm going to give you the long answer, but you can shorten this if it's a one marker. So a short section of DNA that codes for a specific sequence of amino acids. He's run out of room. So that should say a short section of DNA that codes for a specific sequence of amino acids that make a specific protein. That was disgusting, foul, ugly. Ugh. So a short section of DNA is called a gene, 1 mark. You might also be asked, for 1 mark, what does DNA code for? What does a gene code for? And you just need to say a protein. If it's 1 mark, a gene codes for a protein, a specific protein. If it's a 2 or 3 marker and it says explain how a gene codes for a protein or explain the genetic code, then you would say a short section of DNA is called a gene. It codes for an order of amino acids that make a specific protein. So it would run like that. Short section code, specific amino acids, makes a particular protein. Now, you're also going to be asked, whether you're combined or triple, you will be asked about the genome. So what is the genome? The genome is all the genes in an organism. So we've already found out all the genes in a human. We all have the same genes, by the way. We all have the same code. There's variation in the types. So some of us have green eyes, blue eyes, brown eyes, but we all have a code for protein in our eyes. And depending on how much we have, they're either green, brown, or blue, depending on the light that hits them. So we all have eye protein. We all have a protein in our skin as well. We all have the gene that codes for it, and a tongue and 2 eyes and to have 2 ears. We all have a gene that tells us to have 8 fingers and 2 thumbs. We all have those genes. So it's all the genes that a human has would be the human genome. Now, how has the genome helped? This is what you need to be thinking about. It's helped us with migration patterns. So I can look up my ancestors and find that my ancestors 30,000 years ago may have come down from Russia as a tribe and may have moved through India and crossed to Europe, or my ancestors and people from India can trace their ancestry back to, China, Russia, Europe, and it's all about how humans moved about. We can actually see the DNA patterns. How do we know that? Because we find genes, specific sequences of code that are from people from those areas, and you have them in your pattern. They can literally trace where your ancestors went and where they moved about the earth. So the genome has helped us trace migration patterns. That often comes up, like the way where people have moved. Also, the genome can help us with learning about inherited diseases. So we can look at, obviously, the code, and we can find out how these inherited diseases are passed on and stuff like that. There's a couple of other things as well. I can't remember what else, but that'll do. Learning about inherited diseases, migration patterns, education, and there's loads of other future technologies probably from the genome as well. So what I need to do is, oh my gosh, I'm going to give you some questions on this in a minute, but I just need to do, oh my gosh, I just need to do about 4,852,567,8,9,10,11,12 things. Oh my gosh. Right. Let's have a look at, I might come back to the triple stuff. I'm going to come back to protein synthesis and dismiss. I'm going to come back to protein synthesis and mutations. So I need to write that down because that's triple stuff. I'm going to come back to that. Oh, Wilbur Prisk, if you insist. If you're AQA, there's hardly anything to know, but I will go through protein synthesis. Dead easy if you're AQA. If you're AQA, it's a piece of P. So this is protein synthesis, and this is only for the triples. So protein synthesis for AQA, I'm not doing it for the others because it take me too long. So all you need to know is you have your DNA here, right? It's in your nucleus. This is like your holy book. It's got all of your codes and instructions. You ain't letting it out the safe. It's in the nucleus. It's protected, right? What we're going to do, we're going to make a copy of it. So we make a copy of just the section we want. So if I need to make more saliva in my mouth because I need to digest stuff and I can smell some nice cooking, so my mouth starts producing saliva. Saliva is a protein. I need to produce, it contains amylase, the enzyme, the protein, and I need to make loads of amylase so I can digest whatever I'm going to eat. How do we get that instruction out to make that protein? Well, we create a copy of the gene called mRNA. So protein synthesis, a copy of a gene is made. This is called, a copy of a gene is made. This is called transcription, but you don't need to know that. A copy of a gene is made from the DNA in nucleus, called mRNA. Remember, this code is for one protein and one protein only. So this bit of DNA here, you've got 46 of them because 46 chromosomes in your cell nucleus, and you've got one of these in each chromosome. So DNA is in a chromosome. Now, that might have 1,000 genes on there, 1,000 bits of code for 1,000 different proteins. Probably more than that. Probably more like 10,000 different proteins can be made from this one DNA. So we're just copying one bit of instructions, which may be thousands of ATGCs long, a specific code, and that is called mRNA. Now, the mRNA... I found my glasses. Woo! The mRNA now goes and attaches to a ribosome. So here is the ribosome. So mRNA leaves nucleus via something called a nuclear pore. You don't need to know that. Just leaves nucleus, and I'm going to say attaches to ribosome. We all know ribosomes are part of protein synthesis. So there is a specific code on here, and guess what happens? Again, you don't need to know the name of this. Its real name is tRNA, but in AQA we just call it a carrier molecule. So it actually looks like a little leaf with 3 leaflets or whatever they're called on it. Anyway, this is called a carrier molecule. So we're now going to say a bet you're shaking your hand in your English exam. That's what I feel like back in my English GCSE, shaking my hand again from writing too much. Right, a carrier molecule brings a specific amino acid to the ribosome or to the mRNA, I might say on ribosome, oh my gosh, based on the code, right? So you can shorten this down into mRNA is a copy of a gene on DNA. mRNA goes to the ribosome. A carrier molecule brings a specific amino acid. This is the carrier molecule. Here is my amino acid. So imagine there's all different types of amino acids we could bring. This one says, oh look, the code is A, G, C. It says I'm going to bring down the amino acid that goes with AGC. Let's say the next bit of code is C, C, G. Then another carrier molecule will come and it will say, hold on, mate, there's a CCG down there. I'm bringing this amino acid. So the amino acids are brought down in the order that is specific to the code on the mRNA, and then they form a protein, and voila, you have protein synthesis. So we just want to say a carrier molecule brings specific amino acids to the mRNA on the ribosome based on the code, until the protein is made or whatever. There's actually something called a stop code, AUG, that like when it reaches that and you get the AUG, it will detach, and the string of amino acids will detach, and then they curl up and form their 3D shape, become an enzyme. Now, leading on from this is triple. This is triple. No, I didn't want to do that. I wanted a new one, not to copy it, mate. Right, this is triple. So, leading on from this, we're going to see what kind of problems you can have. I'm going to go very quickly through mutations. So, mutations. This is triple. 3 types. You can have insertion or deletion or substitution. What is a mutation? A mutation is a change. Oh my gosh, that is disgusting. Change in a gene. So specifically, it's a change in the code. So it's a change in the code for those amino acids. Uh-oh, disaster can strike. So why will disaster strike? Insertion or deletion, and I could go into it, but it's long. I could show you this, but it will take me too long, and it's just not worth it. Kind of like, you know, if we're doing efficiency of revision time. Insertion and deletion always result in a different order of amino acids. These are much more likely to give you a mutation. Substitution, it's like 50-50. Okay, so what do I mean by that? Let's look at the insertion and deletion, right? Let's say my code is A, G, G, C, T, G. Let's say that's my code. It's just 2 amino acids, right? Actually, let's make it 3 amino acids. A, G, G. Let's say I've got 3 amino acids. Let's insert one. No, diddy. What we're going to do here is I'm going to insert an A there. Guess what? It's called the triplet code for a reason. That is going to flop shot things. So, in fact, I'm not going to insert one there. Let me just, because that I can't even see that, what I've done there. That's just dumb. Let's say I slap an extra A here. Well, we've got all sorts of problems, because now it's going to be read AGG, ACT, and I'm going to delete that there. See, this was AGG. This one at the back was called AGG. We could delete that there because it's read in 3s. It's now going to be read GAG. So all of the amino acids after here have changed. What does that mean? My protein may have looked like this. My protein may have been like this, and it may have arranged itself into this enzyme with this active site because it had this order of amino acids. Now I've changed the order of amino acids. My protein is going to arrange itself with a different shaped active site. So now the active site's completely changed shape. It no longer locks with the substrate. It doesn't work. Remember, all enzymes are proteins. All hormones are proteins. So that's where you get your mutation, and your insulin might not work or whatever, or one of your hormones might not work. Now let's look at substitution. Sometimes we call these can be silent mutations. What the hell is a silent mutation? Well, we can go from ATG GCC. Let's just imagine I change that's C there, and we make that a G. So it went from ATG, let me just show you that as the original, ATG GCC. So I have substituted one of these. One of them has got swapped and incorrectly put together. But that might not matter if the code GCC gives me this amino acid, and if the code GGC also gives me that amino acid, then it makes no blooming difference. The code has changed, but it is still read as that amino acid, so my enzyme or my hormone will still have the same 3D shape and still be functional. So that's why we say that they can be silent, whereas insertion or deletion change all of the code after the insertion or deletion. So thousands of codes change, you're going to get all different amino acids. Anywho, enough on the old mutation, but you need to know that mutations can result in a change in the shape of a protein. Now, most of the time, that's not going to be good, right? If you change the active site of amylase enzyme, you can no longer break down starch, you're kind of screwed, aren't you? Because you can't access the energy from starch. If you have a mutation in your lactose enzyme, which we know a lot of people do, then they're lactose intolerant. They can't drink that sugar with milk. Okay, so if you have mutations in different things, it can be bad. But guess what? Sometimes the change in the protein can make the enzyme fit better and work better than everybody else's. Or sometimes the change in the hormone makes it work better. Or sometimes the change, another protein is in your immune system, antibodies. Sometimes your antibody has had a mutation that makes it fit and destroy the pathogens better. So not all mutations are bad. Most of them don't do anything. Some are negative and some are beneficial. Anywho, let's move on. No, I don't want to get to him yet. We don't want to get to Mendel. What I want to do very quickly is, you may be asked a question on sexual reproduction versus asexual reproduction. So you might be asked about sexual reproduction versus asexual reproduction. You're going to need to know three good things or three things about each. There are advantages. So this is, you need two parents to have sex. It's going to have gametes, male and female gametes. What's a gamete? It's a sex cell, okay, like sperm and egg, pollen and egg, etc. if you're a plant. So two parents, male and female gametes. You do meiosis. When you do cell division, you do meiosis. You get four offspring. I'm not going to compare meiosis. Offspring are genetically non-identical. Okay, you have something called variation. Now, if I wanted to talk about that, having two parents is difficult. You've got to find someone to go out with. If you're a female dog, you've got to find you get the idea. If you're a female butterfly, you find a male butterfly, you've got to have sex. That's difficult. You've got to attract each other. You've got to make a smell. You've got to put colours on. If you're the male peacock, you've got to put all these colours on your back. You've got to parade around doing that all day. If you're a bird, you've got to sing a song. You've got to do all of this to attract a mate. So there's a lot of effort and energy wasted to try and get a mate. And then, what's the benefit of all that effort and energy? The benefit is variation in the offspring, more likely to survive a change in the environment like a new disease. Asexual reproduction is faster. You only have one parent, so you can just they can just do it by themselves. They don't need to find anyone else. Don't have to find a mate. Less energy. You need to know that this is less energy, and it's quicker. It's faster because of that. No gametes. There's no fertilization. No sperm and egg, nothing like that. One parent, no gametes, but we need to know that offspring are identical. Now, is that a bad thing? No. Like, if you've got wicked genes, really good genes, then having identical offspring that also have your awesome genes is going to be great, right? If you have awesome genes in your offspring, that's great. The problem is, if a disease comes along and you don't have the gene or the allele to protect you, then all of the offspring are going to die and be wiped out. So variation means some of your offspring more likely to survive. And that is that. Right, let's move on. Right. What else? I will give you questions on this in a minute. I just want to keep going through the old theory first for a little bit. You do need to know a little bit about meiosis, so I'll go over meiosis very quickly. Some organisms do both methods, and then we're going to do the Punnett squares, the dreaded Punnett squares. So I might just, he says, go on. Okay, let me hit you with meiosis first. So meiosis, I think of the E as eggs, oh sugar, and I don't know, the S as sperm. So meiosis is cell division that makes gametes, nothing else. Only in a man's testicles. Right now, if you're a male, it's happening in your testicles, meiosis. Right now, if you're female, it's happening in your ovaries, meiosis. So in your ovaries, you make eggs. In your testes, if you're a man, you make sperm. Nowhere else in your body does meiosis happen. Nowhere. If you get pregnant, it doesn't happen with the embryo. It doesn't happen there. Meiosis only happens in your ovaries to make eggs and only in your testes to make sperm. Keep that in your head and you won't get tricked up. Right, now, what is meiosis? What's the difference? During meiosis, it has 2 cell divisions. So if you compare that to mitosis, it only has 1. So it has 2 cell divisions, 4 daughter cells, and the daughter cells are haploid. They have half the number of chromosomes, and daughter cells non-identical. And that is it. Let's go daughter cells or new cells. You don't have to call them daughter cells. There you go. And that's all you've got to remember for meiosis. Mitosis has 1 division. You get 2 identical daughter cells and they're diploid. All right, I don't want to spend too long on that. What I want to do next is just flip to the old Punnett squares and stuff. So let me get into the Punnett square stuff, and then we'll get on with Mendel, Carl Woese, and Charles Darwin, and natural selection, fossils, and extinction to wrap up this topic. And then it's definitely coffee break before I do ecology. So, oh my days, yeah. So coffee break after that. Right, let's do Punnett squares and stuff. So first of all, just a quick reminder: sperm is haploid. Eggs are haploid. They get together and make a zygote that is diploid, and then that becomes an embryo, right? And this, folks, if you are asked, it's where people get screwy. Because they see the eggs and sperm, they might be asked what is happening here, and they write meiosis. No, mitosis. These are identical copies in the embryo. So watch out for that. That's mitosis. It's the process that made this that is meiosis. It's the process that made that is meiosis. Everything else in the body, from the zygote to the embryo to the fetus to whatever, is mitosis. Anywho, what I wanted to get down to is actually you can calculate the chance of you having a boy and a girl, and we use this thing called a Punnett square. I like to colour out the first square. You make a noughts and crosses board, you colour out the first square. You're then going to put XX. You do need to know that if you have an XX chromosome, you are female. If you have an XY, you are male. Now XY, and all we do is we go XX makes XX, XX makes XX, XY makes that, XY makes that. X always comes first. So 50% chance boy or girl. Now why have I done that? Because you're going to get asked other questions where you have to combine that, and I'm just going to quickly give you a little, we're going to go through some genetics crossing here, and so here we go, a little bit of genetic crossing, then I'll give you some questions on this, and then we'll move on. I'm going to give you some questions in a minute because I've been talking for about 45 minutes without a question for you lot. So, you got something in life called recessive allele. If you are asked, what is the recessive allele? I might have spelt it wrong, forgive me. The recessive allele, you only need, oh no, sugar. Get rid of that, get rid of that. You need both copies for them to be expressed. So recessive allele, you have to have both, little t, little t. You can use any letter. If they want you to use a specific letter, they will tell you. Then we're going to look at the dominant allele. If a disease or a characteristic is controlled by the dominant allele, and you're asked what is a dominant allele, you say you need both copies. No, Sugar Ray Leonard, he's going bonkers. You need one copy. Oh my days, salty hell. You need, by the way, this is combined and triple. You need one copy for it to be expressed. Now there is something called genotype and phenotype, and if you want to get sexy you can say, ooh, I need one copy in the genotype for it to be expressed in the phenotype. Phenotype is what you see. Genotype is the two alleles you have for any characteristic. So dominant allele, you're going to get it if you've got big T, or you're going to get it if you've got big T, little t, meaning you only need one copy like that and you'll still get it. So let's have a little look how this works. On a real basic level, we can say, let's go basic and then we'll get into cystic fibrosis and Huntington's, and polydactyl is the ones really that they use in AQA. So if you have, big B equals brown eyes, then we would say that is dominant over blue eyes. So what we would say here is, if dad is big B, and mum is little b, look, that person has brown eyes. This person has brown eyes. Little b. Now you always put the big B first. In fact, all of the children have brown eyes, and that is what Mendel found. This is how Mendel actually worked this out. He didn't do it with people, he did it with pea plants and purple and white plants, and he found that purple dominated white. When he bred a brown eye with a blue eye, yeah, but there you go. So that's how you do the crossing. What I want to do is just show you now. Hopefully you're all good with that. I'm going to give you a question now for you guys to do. So here we go. Harry I'm going to give you a question now. So this is going to be a 6 marker. So if you're a true bad man or bad girl, or if you're actually just good at science, you're going to get this right. So 6 fat marks, type of thing that you need to be ready to get. So here we go. Let's say cystic fibrosis. This is the kind of write-up they're going to give you in the questions. Cystic fibrosis is a disorder caused by a recessive allele. What does it do? It makes your lungs fill with mucus, it's harder to breathe, all of that kind of stuff. Anyway, I'm not going to give you that bit of the write-up, but there we go. They might give you a bit of fluff about cystic fibrosis and what it does. Now what we're going to say is Harry and Sally do not have cystic fibrosis. Their son, Jed, has cystic, oh my gosh, this couldn't get any longer, cystic fibrosis. Calculate the probability that the next child they have OMG will be a girl with, oh my gosh, my arm is dying. OMG. Okay, folks, see if you can do this one. This one would be a 6 marker in AQA. It's come up 2 or 3 times before, so this could well come up tomorrow. There you go, 6 marker. Harry and Sally do not have cystic fibrosis, but their son Jed has cystic fibrosis. Calculate the probability that the next child will be a girl with cystic fibrosis. I didn't have to tell you it was recessive. You could work it out from my instruction there, and I'll go into that in a minute. I'm going to give you 2 minutes, see if you can work out the percentage. And yes, what I'm going to do is just quickly grab myself another drink. Have a go at that one. By the way, this is combined and triple 6 marker. Let's go.
Stick around.
I'm gonna have to science the shit out of this.
Oh my gosh, it is so hot in here. So to open my window. Right, let's go. By the way, I haven't looked at the chat in ages, so I'll deal with the chat in a little bit in 5 minutes. Let's get through this question. Where were my glasses? Hold up. I just had my glasses come back in the action. Yes. I can see. Hallelujah. Right. Okay. Like some of these answers, I can see some people have got it. Give a big boom to some people. Right, this is how you work it out, and this is a 6 marker. This is what you're likely to get. Listen, this is how you're going to have to work this one out. You're going to have to go, right, I'm doing a Punnett square, and I'm going to show you how to maximize your marks on this one. Listen, I told you it was recessive, so come on, T, use that. Right, I told you it was recessive. That means it's like on the little t. Now, I don't have to use t, but it's good to use something where the capitals are different. You could have worked out as a recessive, because if Harry and Sally do not have the disease, if it's a dominant disease, one found on the dominant allele, then it's impossible for 2 parents without the disease to have a kid with the disease. Because if one of them had one of the dominant alleles, they would have the disease. So the fact I said they didn't have the disease meant they were what we call carriers. And carriers mean they are heterozygous. So big T, little t, they are heterozygotes. So they are carriers. You can only be a carrier with a recessive disease. You can be heterozygous for a dominant disease, but you will have the disease or the disorder or the characteristic. So we know they don't have it, but we know they must be big T, little t. Because if one of them was big T, all of their children would have a big T and they couldn't have the disease. So there has to be a situation where Jed, who has cystic fibrosis, was homo-recessive. So I know mum and dad had to have a little t each to make Jeddy boy. So let's go and do Jed. Jed, he's got the disease. I want to tell the examiner I know this, and so I beg you, circle it in the exam, because, and I'm going to show you why you will lose 1 easy mark. Because now if you do this, what's that? That's big T, big T. We know they don't have the disease, but the examiner doesn't know that you don't know that. So you need to circle that and say, yes, that's the one that I worked it out from. And then these guys are going to be big T, little t. So it's 25%. Hold on, those people who put 25%, you were wrong. You could still get 4 marks out the 6. 25% or 0.25 that they got cystic fibrosis. But we added in the girl thing. Now, when you have a girl, you're going to do that thing. And we're going to do that. And we're going to go, oh, it's 50%. So I need to go 25 times 0.5 or 0.5 times 0.25. And the answer, of course, can be half of 0.25. What's that? 0.125, 12.5%. Yeah, it's going to be half. So the chance of it being a girl, we have to add the two together and times the probabilities. Boom! Doesn't matter the letters you use unless they ask you. I could go and show you some, but I don't know if I don't know if I downloaded any that are like that. I don't know if I don't think I did. I don't think I did. I didn't do that there like that. But I do need to go through some pedigree diagrams and stuff with you. So at some point I will, if I've got time. I'm smashing through, but if I have time, I will go for. In fact, no, let me just show you now on the whiteboard. It would be much better to actually give you a question, but I didn't have that kind of time to go ahead and prep all of that business beforehand. So let me just show you what's called a pedigree diagram. Okay, this is a pedigree diagram. So let me show you what a pedigree diagram looks like. Circles are normally always girl. They're not going to say girl, they're going to say female, but you get the idea. Girl without disorder. It doesn't have to be a bad thing like a disorder. It could be something else that's genetic. This is going to be boy. I could put normal, but I'm just like, you know, longing it like an idiot. And then that is going to be, I'm going to call this disease X because I'm making this crap up. So girl with disease X. And then boys are normally square. A boy. Again, it's going to be male and female. You're not going to call people boys and girls. It's like very unscientific. Boy, disease X. So you get the idea. If it's coloured in, they have the disease. If it's white, they don't. So check this. I'm going to show you just a little bit of this one. And you're going to, and then you're going to, this is the kind of question they're going to give you. And it's going to baffle loads of people. And we're going to go like this. And we're going to go like that. Look, A and B. What does that mean in one of these diagrams? That means they got it on. And they had some children. And they had C, D, and E. Ta-da! And no, it's going to be much bigger than this, and you're going to have choices because the C is going to meet up with Jim. She married Jim, who's going to be like F. And then they got together and had kid, whatever. You get the idea. It's going to go on and on and on. But actually, I wouldn't join them together because that looks like there's a brothers and sister action going on, and that is not allowed. So that wouldn't be like that. That would be a separate thing, just showing that they are married. So it would be like this. Be like C and F, and then they go on to have their kids, and we'd show their kids. Right? Now, what I want to say to you is, how do I know... And this will be 2 or 3 marks. So big fat question in the grand scheme of things. How do I know disease X is caused by a recessive allele? I meant to say cause is caused by recessive allele. Who can tell me how I know? Evidence. They will say give evidence. Disease X is caused by a recessive allele. Have a look. A and B do not have the disease. They have had a child with the disease. What does that tell you about A and B? If you think about the letters we use, what does that tell you? A and B must both be heterozygous carriers. Excellente, Emmanuel. As the parents don't have the disease and have had a child with the disease, it must be recessive. Listen, if the disease was, look, you've got 2 alleles. I'm using big T, little t. If the disease was found on the dominant allele, then that would mean that for A and B not to have it. So if it was a dominant disease, A and B would have to be like that. Because if they had 1 big T, they got the disease. Now it would be impossible for them to have a kid with the disease because all of their children would be lower T. So it cannot be a dominant disease. So they could say, give the evidence this is not a disease caused by a dominant allele. And you'd say, well, A and B, you know, you get the coup, right? For them to have had a child with the disease, it means they must be big T, little t, and that one must be little t, right? And the disease must be found there. That's the only way this thing can work. So another thing they could have asked us is, what is the evidence Another question, what is the evidence that B, they're not going to ask like this, not going to say, how come B heterozygous? They're going to say, give the evidence why B is heterozygous. And you're going to say the same exact answer. You're going to say, A and B had a child with the disease. A and B must be carriers. Recessive illness, they must be carriers. Therefore, they must be heterozygous. Each one must have had the recessive allele. Yeah? They can't be homozygous recessive. We know that B can't be little t, because if B was little t, they'd be coloured in. They'd have the disease, and they don't. So we know that they can't be little t. We know that they had a kid with the disease, and you get one from mum and one from dad. So they had, and the kid C had little t, so we know they had to be hetero. Okay, anyway, enough of that business. I'm going to move on. And this is Mendel. What do you need to know about Mendel? Very, very quickly, I'm going to tell you 3 things about Mendel, and then we're going to move on. So Mendel. He was a monk. I think he was Austrian, German or Austrian, one of the two. Gregor. And his name was Mendel. And what did he do? Well, he couldn't have sex himself because he was a monk. And so he would have had a vow of celibacy. Not all monks do, but I think he probably did. And so then he got a bit excited. And so his favourite pastime was to reproduce plants. So get plants to reproduce. And what he found out, because he was a clever dude, but none of his discoveries were found until after he was dead. And then people found his books that he'd written when he was in the monastery, in the gardens, looking after the pea plants. And they were like, wow, this dude found some stuff out. So what did he do? He got purple plants, dominant purple plants that now no one had heard of a gene, but basically he bred the plants 3-4 years in a row and they were always purple. So we had purple plants. He knew they were purple. He took the purple plants and he mixed them with the white plants that he'd had for years, and he knew they were always white. And what did the kids turn up like? All of the kids, however many kids there were, however many offspring, they were all purple. So this made him realize, he came up with the fact that these are the things he discovered. There are units. Now, he didn't use the word gene, but he said there are units of hereditary. I don't know how to spell that. Units of inheritance. I was better off saying inheritance because I can't spell that. So he basically discovered genes, even though they weren't called genes. He'd never seen genes. He just knew that something is being passed on. How did he know something's being passed on? Because every time the purple ones, and then when he got the purple and white together, all of their kids were purple. And then he also discovered some units are dominant. So he discovered some are dominant and some are recessive. How did he know that? Because he was like, listen, I mixed the white and purple plants pollen like this together, and all of the kids came out purple. That means whatever decides the purple colour is dominant over what decides the white colour. So he also found that out from his experiments. And the last thing we need to know about him, of course, he then did the kids. I'd say those are the 2 main things. There are units of hereditary and some units are dominant and recessive. There we go. I'll not spend too long on him. Right, yeah. When he did the kids, he found that some of them were other colours and whatnot. So that is Gregor Mendel. You might get a 1 marker, blah, blah, blah. I don't know what I'm waffling about. Where am I? I'm losing the wheel here. Let's go. Come on, speed it up, mate. Speed it up. Sort it out, guys. Oh my God, you made a circle. What a twit. Okay, who is this? This is Charles Darwin. So Charles Darwin, him and a guy called Alfred Wallace worked together, actually. Alfred Wallace, I think, was in Australia, and they wrote lots of letters. Back in them, no one had text message, so they had to write letters to each other, and they kind of both explained their theories, and they had similar theories. Charles Darwin spent years and years writing a book, but he sat on it for like 20 years and didn't publish it. He was too chicken because it was such a shift in the way of thinking. But how did he come up with the theory of natural selection? These guys came up with the theory of natural selection. And something you've got to know about natural selection, I'm going to explain how their theory works. It took him years to publish his results, but he came up with it because he was a creative thinker and he could think outside the box. And it's those people with that kind of intelligence that make the huge genius level breakthroughs that other people can't make, which shift the entire way the world thinks about a problem. So yes, he was a definite genius, and it's because his brain did not work in the same way as everybody else's brain, and that is what happens with geniuses. So now look at these zebras. Some of these zebras don't have stripes and some have stripes. So we're going to go and explain why do all zebras have stripes, and this is by natural selection. So I'm going to run through this in bullet point form for you. You need to know these 5 bullet points. So what we want to say is variation. Now, if I've got a question about zebras, I'm going to say some have stripes, some don't, bruv. That's my variation. But I'm going to say variation is caused by mutations. Now, if you want, you can say a mutation is a change in the genetic code of a gene or of DNA. Lovely jubbly. We just did that with the different mutations. This is for triple and for combined. So variation is caused by mutations. Then what we want to say is this new gene or new alleles, you might end up with a new allele or new gene, some new or mutations. Let's say mutations. Let's stick to the mutation. A mutation is just a word for a change in a gene, and it doesn't have to be bad. So some mutations give beneficial. What does beneficial mean? Beneficial means good. What's a characteristic? Well, the zebra has stripes. Another characteristic, we might have honed in on its ears if I wanted to explain why it has these ears. Everything on your body can be explained by natural selection by this 5 marker, okay? Everything in the world out there can be explained. Why do cactus have thin leaves? Why do, I don't know, dogs have big ears? Or why does a fox have a bushy tail? Anything you come across, it doesn't matter the context, it follows this. Variation is caused by mutations. Some mutations give beneficial characteristics. These make those individuals with the beneficial characteristics, so I'm just going to say these make individuals more likely to survive. You've heard of survival of the fittest. Now, are they asking you to know exactly how stripes make a zebra survive? No. They want this mechanism. Of course, it might be that when lions, wouldn't be a tiger, would it? Don't live in the same place. But when a lion sees a zebra, it might be that the stripes confuse them and they can't see what's the head, the tail. When you have 50 zebra together, the stripes is like, whoa, man. And it freaks them out. Whatever. These make the individuals more likely to survive, reproduce, pass on genes. If you were doing A level, you'd say alleles, but GCSE you could say pass on genes to offspring. And then we might want to say this happens many times, happens over many generations. And that is how zebras end up with stripes. It would have been one freaky striped one and all the others going, look at this idiot over here with the stripes, man. What a plum. Of course, that one survived. Everyone else is getting eaten. That one survived, passed on its genes to the offspring. Now they were like 4 stripy ones in the group of 1,000. They were like, look at these freaks. And then, half of them get eaten again. And the 4 stripy ones survive, have sex. Each one of them has kids. Now there's 16 stripy ones in the next generation. Still more of the plain ones. And then that carried on going on and on and on. And after many generations, they've all got stripes because the stripes gave them maybe a 2% mathematical advantage to survive being eaten. And that is why when we look at them, they're all there. So variation is caused by mutations. Some mutations are beneficial. These make you more likely to survive, have sex, reproduce, breed, whatever word you like, pass your gene on to the offspring. Happens many times. That is natural selection. That was Darwin's theory. Now, don't slightly different for speciation. 1549. Yes, you have a question. Can we do the melt practical now? Wilbur, I didn't ask. No, I will do the melt practical. I'm not going to do it just yet. No. So I'm kind of going in a rough order here of things. The melt practical is going to be in the ecology section. OMG, I'm going to be going till 10. So I'm going to be going till 10. If I get there, I get there. If I get there, I have a very, very bad habit of waffling on, I know. But you know what? Try to teach the whole of the GCSE in 4 hours. It is hard. If it could be done, what would be the point in having, in the whole of year 10 and year 11, having 200 hours, eh? If it could be done, then you wouldn't have 200 hours of lessons, 189 days in the year, 39 weeks in a school year. Most of you, if you're in year 11, have had 2 hours a week of bio. So if you go 39 times 2 for year 10 and 39 times 2 for year 11, you've had just under 160 hours of biology. I try to teach you it in 4 hours. Very difficult. All right, I'm trying to do 80 hours in 4 hours because we're doing just B2, so I don't want to get too excited. But speciation, what is that about, right? That means there was an ancestor. There was a bear, right? Here's my ancestor. It's now extinct. How does one, this is slightly different than natural selection. It's on the same thing, but how does one thing become two different species? So this is what we want to say. I'm going to narrow it down for you. We want to say isolation. What does isolation mean? We don't care the reason. Oh my God. Right. Isolation, it can be, it's called reproductive isolation. They would have both been like this color. I'm making this up, by the way. They would have been like a certain color. They got separated by mountains. The tribe, the bear crew were going. Half of them went down there. The other half said, bro, I'm not going down there. The fish are rubbish down there. I'm going down here. They went down here and no longer could they climb the mountains, you know, due to whatever natural changes. So they were stuck on either side of the mountains. Isolation, we call this. Now, reproductive, you can, this particular type of isolation, that means you can't have sex, it's called geographical isolation because you can't cross the mountains. So it's a barrier, and you can talk about a barrier, a river, an ocean between islands, anything that stops the two populations getting together. So we have population A, we have population B. They would have looked the same and been the same, but the environments are different. You can clearly see that one is covered in snow, one is a little bit warmer. So we have environments are different. And then all I have to say is natural, unless it's like a 6 marker, then if it's a 6 marker, I'll bust out a bit, I'll bust out the natural selection 5 point. But otherwise I'm just going to say natural selection occurs differently, right? So remember I was talking about the stripes on the zebras? Well, here it's going to be different. If you were brown over here, you stand out. And every time you go to eat a seal, he goes, oh, look at this joker, he's coming, right? And the light one, that was a bit freaky and maybe like the weirdo in the tribe, everyone was like, hey, mate, you're light. Look at you, you weirdo. He actually was like, yeah, I'm getting all the food. These idiots can't catch the seals. So now, does that mean that he was a white one? No, just slightly lighter meant he had a slightly, could get a couple of meters closer, and over time those ones had a higher probability of surviving and passing on their alleles. Anyway, natural selection occurs differently. I don't know what difference means, but occurs differently because of the environments. Okay, so different alleles, different genes are favored. Now over time, we can say after many generations, a generation is like many reproductive cycles, many lots of kids. After many generations, if A and B meet up, so let's imagine a human takes B, brings it to A, and they get it on, right? If A and B meet up, they can reproduce, they can have sex, but they can no longer, I need to change this, they can no longer produce what we call fertile offspring. And this comes to another point. You guys have to know the definition of a species. The definition of a species, if a scientist wants to find out, is the brown bear and the white bear there related? Can they have sex and have fertile offspring? So if A and B meet up, they can no longer have sex and produce fertile offspring. Now I should really say reproduce rather than have sex, because sex is a shorten for sexual intercourse. So what I want to say is they can't reproduce and produce fertile offspring. So the definition of a species is you can have sex and have fertile offspring. A lion and a tiger can have sexual intercourse and we get a liger, but the liger can't have kids. There's no species of liger. A horse and a donkey can have sex and they have a kid, we call it a hybrid. It's called a mule, and a mule can't have kids. There's no species of mules. They don't make gametes. They don't make sperm cells. Same with the liger. So tiger, lion have sex. That means once upon a time they had an ancestor that got split up. Tigers you find in India, lions you find in Africa. So once upon a time they had an ancestor that got split up. Okay. And now we know they're related because they can still have children, but those children are infertile. Anywho, that is known as speciation. Yes, that's the process of speciation. I've done insertion, deletion, and substitution. Substitutions can be silent. Insertion and deletion always cause changes in the protein. What I want to go through quickly, a couple of other things in this topic. So I'm going to get through this topic and then I'm going to take a quick break. I'm going to give you a load of questions to do. So this is really what is going on here. What is this dude doing? Oh, this is supposed to be selective breeding. So let me go through selective breeding. I mean, we know that natural selection is a pretty cool theory because we can do selective breeding. So all you're going to do is take a male and female, I don't know, gametes from organisms. Oh my gosh, I'm longing this out with desired characteristics. So what am I saying? Let's say I want characteristics. Let's say I want tomatoes that don't get killed by a certain disease. I'm going to take some pollen from one tomato plant that has got really tough breed of tomatoes that don't get killed, and I'm going to take another tomato plant that don't get killed, and I'm going to mix their gametes and make them have children. Okay, so I am taking, if it was a cow and I want it to produce lots of milk, I take a female cow that comes from a line of women, female cows, generation after generation, that produce tons of milk. And then I go to a male cow that also comes from a family of cows that produce loads of milk. I know male cows don't, but you can still take a man from a family of cows that produce lots of milk. So you take that male one and you mix it with the female one. You get them to have sex. It can be difficult if they don't want to have sex, obviously. And you breed them. And then you take their children. And this is the weird bit. You take like the brothers and sisters, you take the offspring and you repeat. So you're going to take the children, and it's kind of like incest. You're taking the children and you're getting them to reproduce. What you're trying to do is strengthen the desired characteristics, and you keep doing it until all of the offspring have the desired characteristic. You do it for many generations. The downside to this is it introduces genetic weakness, a lack of variation in the gene pool, which means they're less likely to survive changes in the environment, and there's a higher chance of inherited disorders. So take a male and female gamete from organisms with desired characteristics, produce offspring, and then we want to say select offspring with desired characteristics and repeat. So it's gonna take you time, because you might want to make your tomatoes disease resistant. You might do that for a certain amount of years, and then you might say, right, now I want to make them big and tasty. So it takes time to do this selective breeding. Select offspring with desired characteristics and repeat. Do many times. When you do it until all of the offspring have the desired characteristic, then you know you're done, right? You've been successful. So what are the downsides? What are the bad sides to this? Obviously, what are the good things? Well, they all get disease resistance, or you get cows that produce loads of milk, or you get really sweet smelling strawberries or lovely tasting strawberries. But the downsides are reduction in variation. So reduction in biodiversity, reduction in variation, less likely to survive change. Oh my gosh, I can't be bothered. Less likely to survive change and more inherited disorders. I'm getting tired of writing. You get the idea. Right, let me move on. Let me move on. I know how you feel. I'm bored too. There we go. Right, you do have to know about cuttings. What are cuttings? Cuttings is cheap and quick. So just make sure you know that cuttings are cheap and quick, and you get identical offspring. Okay, and that is with plants. We can take cuttings. They are what they say on the tin. Cut a bit of the shoot, cut a bit of the root, bosh, and you can grow it. Then we have tissue culture. So I'm going to speed up a little bit because it's taking far too long. You have tissue culture. What is the difference with tissue culture? Tissue culture, you make millions, so you make many more and faster. So it's faster than cuttings. So tissue culture, identical offspring. Again, you're making clones. We call these clones, and it's faster and more, so it's better. What's the downside? It's more expensive. That's the only downside, okay? But you're going to make genetic identical clones faster. Right, let me go through some fossils. Preservation. So 3 different types of fossils that I want to go through. You have our preservation. What happens here? Oxygen cannot get in there. Without no oxygen, so cannot decay. I'm nearly at the end of unit 2. No oxygen, so it can't decay, right? If you can't get oxygen in there, it can't decay. And so the mosquito, that is the best type of fossil you can get. We call it a preservation fossil, okay? And that's the best type of fossil you can get. Why? You can see, look, color. You get to see color, soft flesh. Yeah, you get to see, oh, it's got wings. Oh, look what its color is. You can see all of those things. You cannot see that with the other 2 fossils. So the other 2 types of fossils, this fossil is known as mineralization. You have to know about this. Fossils are evidence for evolution. Mineralization. Fossils are evidence for evolution. The further down in the rock the fossil is, the older it is. So mineralization, you do... Oh! You get no soft parts. All right, you get no soft parts, so you don't know the color. You have to guess the color of the dinosaur. You have to guess the color of its eyes. You do. And what is mineralization? The bones are not left. Hard parts are swapped with minerals. So hard parts are swapped with minerals. So you almost get like the bone. They're not real bones. They're not real bones. You get minerals instead of the bones. The bones over time, over millions of years, swap for various minerals in the environment. And then the third type of fossil that you can get, because there's 3 types. This is probably the least useful, but it's still decent. It's called a cast or an impression. So this might be 1 million years old, this footprint. Now, can I tell the color? No, I don't know what color animal that was. I don't know a lot about it, but maybe I can look at the sharp thing and say, hey, this was a predator, right? But maybe not. Maybe it needs claws to hang on. So maybe it hangs on to trees. I don't know. But we can tell things about it. How many toes it had. Did it have claws? How big was it? How heavy was it? But it's still less information than the other 2. The mineralization, you actually get the structure, but no soft parts. And then the preservation, where you're frozen in ice like woolly mammoths or like mosquitoes trapped in amber. Okay. I haven't gone through dialysis. If I'm still alive at the end, I will. If I'm still alive oh my days, what the bloody hell is this? Woo 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 woo. Yeah, it is easy. It is easy. It is easy. B2's nice and easy, guys. This is just all the I'm just going through all the content. I could have just done exams. I'm always in the thing. Do I just do exam questions? I could have just done hundreds of exam questions. You know that, and it might have prepared you better. But you know, sometimes you know, people want the explanation, and then it involves me talking. It's much easier to just go through the millions of exam questions. I've got a video where I will go through 100 B2 multiple choice questions. You'll find that I've got that. I did that yesterday, 100 B2 multiple choice questions, and went through 100 questions in an hour, so that might be more effective revision, much more effective than this. Anyway, all you need to know for antibiotic resistance, you just need to know mutations. This is antibiotic resistance. If I had to remember anything if I had 1 minute to revise it, if I had antibiotic resistance, you get mutations, make resistant gene. All right, that means that some bacteria have the resistant gene. Guess what? It's exactly the same as natural selection. They survive, reproduce, pass allele, you can say gene, on to offspring. Okay, so essentially, doctors, this came up in B1, doctors shouldn't overprescribe antibiotics. Farmers shouldn't use antibiotics, because what they're doing is they're killing all the bacteria who don't have the mutation, and the ones with the resistant mutation are surviving, reproducing, and now you have a whole population. Instead of having a whole population of zebras with stripes, they're all coming back with resistant genes to antibiotics. So you're wiping out all the ones that don't have it, and then all the ones that survive are resistant, and you end up with superbugs. And one superbug you have to know the name of is MRSA. So leads to superbugs. And superbugs like M to the R to the S to the A, MRSA. Anyway, I'm going to give you some questions, then I'm going to move on. The last bit of this topic before we finish this topic is this dude. There's actually 2 bits to do, 2 last bits. This guy is called Carl. I probably spelled his name wrong. Carl Linnaeus. Okay, I think he was Swedish. Anyway, I think his name was Carl. Jeez, should know. Why have I got this on here? Well, anyone know what this stands for? What did he come up with? He came up with what we call classification. And he basically, King Prawn Curry, that's the one. It's not Carl Woosey. Carl Woos came up with another bit later. Carl Woosey is still alive. Carl Woos came up with stuff in the 1990s. This geezer's died ages ago. This guy was 1800s. So this guy's died ages ago. He was before the First World War. So Carl Linnaeus, he was hundreds of years ago. So Carl Linnaeus said, look, when we classify things, we need to put them into groups. Kingdom, king. Phylum, prawn. Class, curry. Or order. Fat, family. Greasy, genus. Sausages, species. King prawn curry or fat. Greasy, sausages. I nearly wrote sausages there. You don't want to be in the exam and say, yeah, he's a sausage. He might have been a sausage. I don't know. There we go. Kingdom, phylum, class, order, family, genus, species. Now, there's no overlap between these. Now, obviously, that's long. That means that all of us have 7 names. I am animal, because kingdoms, there's plants, animal, fungi, protist, and bacteria. Prokaryote, like bacteria. So that's the 5 kingdoms of life. So I should say my name is animal. My phylum is vertebrate, because I have a spine. My class is whatever the hell, mammal. Mammal is my class. My order is primates, because I'm hairy and I walk around on 2 feet. My family is, I don't know, hominid or something. My genus is Homo and my species is Sapien. So that's a long ass name. So what he said is, you don't need to name every animal with all 7 of them. He said to speed it up, we're going to use something called a binomial naming system. Bi, what does bi mean? Bi means 2. So a 2 name system. So we said we're going to use a binomial naming system, and the binomial naming system works like this. So he used binomial. Nomial means naming, so binomial. And we start with the genus and then the species. So you have your genus and species. So like Panthera leo, and that is a lion, a Panthera leo. Now, Panthera is its genus, so any other cat that has Panthera is in the same genus as the lion. But if you're a Leo, then you can have sex with a lion and have kids with a lion, so you are a lion, right? So only lions are in the same species. Now notice this is lower case. I can't even write that on there. But basically the species, oh jeez Louise, Tom, sort it out, you dipstick. Anyway, I'm not going to write it down, so you just have to remember it. Species is always in lower case, meaning lower letters, and genus always begins with a capital. Boom. Okay, right. Last thing in this topic, then I'm giving you loads of questions, and before I maybe attempt to do ecology, what the arse? So then came another dude. His name was Carl Woose, or Carl Woosey. I think he's Woose, but Carl Woose. Now this was only in the 1990s. What's that? Let's just say only. That's like 30 years ago. So Carl Woose, he said, "Hold on a minute, mate. We've been doing this for far too long." He said, "We've been going king, prawn curry, or fat, greasy sausages. We've had a 7-step naming system." He said there's 8. He said, "You're saying that it starts with, are you an animal or are you a plant?" He said, no. It doesn't start with, are you an animal or are you a plant? It starts with, are you a eukaryote? So do you have a nucleus or not? Or are you a prokaryote? And guess what? Then he started studying prokaryotes, like this one here and this one here. And he looked at their DNA and he found that the old ones, called extremophiles, they love extreme conditions like living in volcanoes. He found these extremophiles and these modern new bacteria. He found that their DNA was so different, he said they have to be put into 2 separate groups. So we have a first layer, and he called it the domains. So it's actually a D, so it should be like deadly king prawn or delicious king prawn curry or fat greasy sausages. So it's actually, he came up with the domains, and this is just to show you that new science, new evidence, due to new technology, because Carl Linnaeus didn't have DNA and we did, he came up with new things. So we got eukaryotes, we have archaea, which are ancient bacteria, and then you can call these prokaryotes or bacteria. Prokaryotes, or you can just call these bacteria. Anywho, that is the end of that unit. I'm going to give you some questions. We're going to go through questions, then I'm going to take a break, then I'm going to do like 40 minutes of ecology, and then I'm going to blow my brains out because I will have been doing the live for like 3 hours and I will be dead. I'm going to do the paper 1 content at 1 a.m. in the morning. If you come back here at 1 a.m. in the morning, I'm doing the paper 1 content. Yeah. Well, I'm going a bit crazy. So potato blight, people. It's a great disease. Probably the best. Right. Potato blight is caused by fungus, Phytophthora infestans. What a beautiful name. What is the genus of the fungus that causes potato blight? Is it the infestans? Is it Phytophthora, or is it Phytophthora infestans? Which one do you reckon, guys? Yeah, it's the second one. How do we know it's the second one? Because the first one in the binomial name is the genus, and it begins with a capital. Look at that, lowercase. That is the species. So if they had said, what is the species? This is the species, this is the genus, and this is the binomial name. Boom! Oh my gosh. Got a few more questions for you. Anybody know what X is?
Stick around.
I'm gonna have to science the shit out of this.
Yep. Oh, yes! Thank you for calling me sugar. Yes, it is sugar. You're right. It's called ribose or pentose sugar. X is sugar. Boom. Right. Let's go to the next one. Next one. We're on the questions. What is the name of the type of cell division that produces little sperm cells? So who's going to produce my little haploid sperm cells? Not my particular ones, because I'm going to produce those, but what process makes sperm cells in the testes of a dude? Well done, guys. It's meiosis. Meiosis, you can think of the E and the S as eggs and sperm. Boom. Right. During meiosis, the cell divides twice. How many sperm cells are formed? Huh? How many sperm cells are formed when a cell divides by meiosis? Huh? So, this is a bit confusing, and I'll go through that. How many body cells contain 46 chromosomes? How many chromosomes are in each human egg cell? Oh, man, you guys are going to get the double boom. Boom. I'm too tired to even give one of my proper booms. Like, boom! Boom! Okay, yeah, absolutely. Yeah, well done, guys. You're going to have 4. Now, that can confuse the crap out of people, so let me just run through it. First of all, it doubles its DNA, right? Yeah, anyway, no, I'm not going to run through it. We don't need to waste time. You're absolutely right. It's going to have 23. Absolutely right. No need for me to talk out my backside. Right, we're going through questions here for Unit 2, and then, oh my gosh, I think I'm just going to go through questions for ecology. I don't think I can go and teach the content. I stupidly tried to teach the content. That's like dumb. I should have just been going through questions. But you know what? You can't be perfect. And I'm far from that. Look at these glasses. Amazing windows. It's just due to coffee and going insane from being up so late. The table below shows part of the classification of the ring-tailed lemur. Kingdom, phylum, genus. Complete the table above. You guys are all on this. Just don't write curry. So king, prawn, class, or fat greasy sausages. So order, family, and species. And this one is who came up with the theory of evolution by natural selection? Tick 2 boxes. I don't know, really. I don't know. I haven't got a favourite film. I think there were. I'm going to be biased. You can't. Everyone's going to be biased for their era. So all my favourite films were in the 1980s. I think the 1980s was amazing. 1970s was quite grim. There's some awesome films from the 1970s like Jaws, and that 1980s had awesome films. I think, you know, if I was writing a top 100 films, loads of them would be 1980s. 1990s had some decent films, but yeah, 1980s had some just incredible films. Like Back to the Future was just like wow. Indiana Jones, you know, and the Temple of Doom, whatever, Raiders of the Lost Ark, Star Wars, although Star Wars was 70s and 80s, Return of the Jedi was 80s. But like there were just so many good films in the 80s. 90s had The Matrix, Pulp Fiction, Reservoir Dogs. Like it had loads of good films as well. You know, recent films, you see, I'm going to be biased. I think all the recent films are trash. They're all just superhero trash, CGI trash. There's nothing original, exciting, or like it's all just boring trash to me. But there we go. That's me. Yeah, I think the best films were 70s, was decent. 80s. I mean, 60s. All of the eras had good films. All the eras have good films. But I mean, I can't think of any films I remember from recent times that I go, wow. Like, whereas in the 1980s, you walk out the cinema and be like, wow. Or like when I saw The Matrix for the first time, I walked out, I was like, wow. You know, like you're walking home, everyone's talking about it. Like, it was heavy. Yeah, 1980s Back to the Future was just awesome when that came out. Right, but there were loads of films like The Terminator that came out in the 1980s. Sorry, that was my Arnold Schwarzenegger impression. Right, yeah, we want Alfred Russel Wallace and Charles Darwin. Yeah, Terminator was good. Aliens, the Alien and the Aliens and Predator and all that came out in the 1980s. Running Man, Total Recall. There were just loads of good action films. And then the 90s had the Die Hard movies. There's loads of good films actually in all eras. But yeah, what is a fossil? OMG, that's a hard question. Give two. I never went over this. That's why doing questions is so much better. Give 2 causes of extinction. So what is the fossil? I'm just gonna say, guys, it's the remains of what? a dead organism. I mean, this particular foxel, I can't even speak. We know what that is. That's we might be asked to name that. 2 causes of extinction. What are 2 ways that people can become extinct? There's like 4 ways you have to know about. So you can say natural disaster, like a volcano, an earthquake. So we can a meteor strike, an asteroid strike that supposedly wiped out the dinosaurs. Natural disaster, we can say. You can also say the word new species, new. Let's put that in a bit more. A new predator. So let's imagine an alien came from outer space and ate all the humans. That would be a new predator. We can deal with all the predators here. So a new predator might sort us out. Competition. You know, if competition, 2 species fight and 1 survives. They don't have to kill each other. One can eat all the other one's food. So competition, yeah. So we've got some ones there that we can go for. Climate change, you could probably go for as well there. How was the fossil figure above? Animal walked on mud. So this is the cast or impression fossil. So if you had to explain that, the animal left a cast or an impression. What the blum? Yeah, so 2 of the following boxes, evidence for evolution. Bacteria can become, well, that proves natural selection, right? That's evidence. Why? Because bacteria can reproduce every 20 minutes. Imagine I wanted to test natural selection in humans. I've got to wait 50 years. I've got to wait 20 years for everyone to have kids and then look at their kids and go, did anything get passed on? Right? Whereas with bacteria, I can wait 20 minutes and then I can examine them and go, oh shit, look, that gene got passed on. So bacteria, and the fact that they become resistant when we give them antibiotics in a dish and we can see, oh look, it's become resistant, we know that it's been passed on. Older fossils are deeper down than newer fossils. Let's check this one. What is the genus of the Siberian rhinoceros? Woo! That's a big horn. Yeah, we'll do some written ones. We'll do some written ones as well. Hopefully, yeah, I'll bust out some written ones. Yeah. So the genus of the Siberian rhinoceros. You might say, how the fudge are we supposed to know? Well, check this, guys. This is going to be binomial because it's 2. That is got lowercase. So if you didn't work it out, that's lowercase letter. So that is the species. So the genus is the one with the capital letter. Boom. Which domain was the Siberian rhinoceros in? Archaeokaryota or prokaryota? So what do we reckon? It's a eukaryote. It's got a nucleus. It's an animal, right? So it's part of that. Right. Who developed the 3-domain system? Carl Woese. We went over this. You should know this. Right, what I will do, let me get up. Somebody said, can we do some longer questions on this Poopa Loopa? Do you want me to move on to ecology, or shall we do ecology questions, or do you want to do more on, oh, give me 1 sec, folks. Give me 2 minutes. All right. Let me zing on to some ecology questions. I'm just going to find a couple of 6 markers. See if you can do that. See if you can do that. Do that. And then I'll find some 6 markers. Right. I'm going bananas, so I might talk a lot of fart in the next few minutes. I'm going to try and get up some 6 markers and stuff. So here we go. Let's go. What is that? That is inheritance, variation, and evolution. Hold on, let me give you a couple of 6s. Do you want to just do ecology? Someone said a 6 marker on fossils. Good gosh. Oh my gosh. Oh my god, some of these are disgusting. Oh my god. Oh my lord. Just remember that with your fossils, they get destroyed and stuff like that. Right, let me go through these and then I'll bust out some 6 markers. Let me go through these. Could you do some ecology content, please? Yes, I can go through the ecology. So first of all, we have abiotic and biotic factors. What does this mean? Biotic is living. Abiotic is non-living, right? So we have living factors and non-living factors. Which ones of these are living things? Well, nitrates in the soil, that is a non-living. Rabbits eating plants is biotic. Shading by a building, that's a non-living thing. A building's not alive. Whether the soil is acid or alkali, temperature, these are all non-living. Trampling by people is biotic. So all of these are going to affect the population. Now, you've got some definitions you've got to know. So here are some definitions you have to know. Habitat, population, community, abiotic, biotic factors. We already did this one. So abiotic factors are living factors. Abiotic factors, oh my gosh, is non-living factors. Come on, Tom, where's your pen, mate? Habitat, what the hell is a habitat? That's somewhere where an organism lives. And then we've got ecosystems. Ecosystem is everything. All the populations of all the different... No. Oh, right. That is the ecosystem. So it's all of the community, by the way, means all of the populations. So where is that? All the populations of all the different species. That's called the community. And a population is just one species. So all of the individuals in one species. But a community is all the species, and ecosystems is everything. It's all the animals, all the plants, and then it's the non-living factors, the sunlight, the water, the temperature. So like the desert is an ecosystem. We'd say the cacti is part of the desert ecosystem. Camels are part of the desert ecosystem. The sand, which isn't alive, is part of the desert ecosystem. So it's all the living and non-living factors in one area. Okay. Now, you do need to know what are 4 things that animals compete for. And of course, these are things that can cause extinction as well. And you have to know that animals fight for food. They fight for mates, i.e. who they can have nookie with. So food, mates, who they can sexually reproduce with. I don't know, water, shelter to bring up their kids, or territory. You can call that territory as like an area to protect. What do plants compete for? Well, plants compete for water. Think about photosynthesis, sunlight. Do they have to fight over carbon dioxide? No, they don't have to fight over carbon dioxide. Space for roots. So if 2 plants are together, they'll be like, I'm putting my roots there, whatever. So they're going to fight over their roots, right? Space for roots. Water, sunlight. We could say stuff from the soil like minerals, etc. Yeah, let me just leave it at that, like magnesium that makes chlorophyll. So they might fight over that, or nitrates. I'll leave it at that, leave it at that, leave it at that. Oh my god. Right, what else? Give me 2 shakes of a cat's tail. I think over genetic engineering, never mind. Louder. Things pop up in my head that I didn't mention. 3 types of adaptations. An adaptation is a feature or a characteristic you have that helps you survive. There can be structural, behavioral, or functional. Structural is anything physically that's different on the body. Like I have big ears to let heat out, or I have big ears so that I can hear predators coming to eat me. Behavioral is like I hunt at night. That's a behavior. All right. I hunt at night. I sleep in the day, so I'm nocturnal. Or I'm diurnal. I hunt in the day and I sleep at night. We're diurnal generally, unless we're revising for our GCSE. And then functional is kind of like related to stuff inside the body. So like if I have a high metabolism, that would be functional, a functional adaptation. Right, let's look at predator and prey graphs and food webs and things like that, and then we'll do some questions on that stuff. So you have food webs, and all food webs start with producers. The arrow shows you the flow of energy, and these would be called primary consumers. Now, primary consumers are herbivores, they're vegetarian. And then this is known as a secondary consumer. Now, he can also be described as a predator. OMG. So I'm very briefly going over this. This is a prey. But this is also a prey. This one is a prey to this bird here. And then you could be asked to give, like, if all the frogs get mashed up, how will that affect the number of snakes? So we could say the snake population will go down. Why will the snake population go down? Because it will have less frogs to eat. Or we could say the snake population will go down because the hawks will have to eat more snakes. Right, you get the idea, right? So you can kind of like go through, but often there's more than one answer you can go with. You can go with either one. As long as you explain it, you can go with either one. Okay, so anywho, that's called a food web, and that shows this word you need to be aware of. So if you've never heard of this word before, you need to be aware of this word in ecology: interdependence. And this means that all the animals within a food web are linked. So if I kill the mice with my poison, because I'm a farmer and I don't want mice eating my rice, so I kill the mice, then the snakes eat more frogs. And if the snakes eat more frogs, then these producers increase and they eat all my bloody crops. Serves me right for killing the mice, right? So there's a link. One thing you do in the food web to one animal in the food web can cause changes in populations of other animals in the food web. Okay. Yeah, that was interdependence. So I went through that. Yeah, good people putting some good knowledge in the chat. Chat. All right, have a go at these ones, guys. I'll put one up on Pete in a second, on old Peter Piper. Yeah, I've gone through some homeo. Ooh, what is that? Is that a chocolate coin or is that a gold coin? Gold coin. Nice. Okay. Name one consumer shown in this diagram. What's a consumer? So you could go with snail because it consumes the lettuce, or you could go with shrew. Either one would be good. Name one carnivore shown in the diagram. One carnivore in the diagram. Well, I can't say snail. I have to say shrew because the snail isn't a meat eater. So remember, carnivore means meat eater. And then a disease kills most of the shrews in the garden, suggests why the number of snails may go down. If a disease kills the shrews, what would happen to the snails? Why would they go down? Hold on. Suggest why the number of snails in the garden, may increase. See, I'm blind, even with glasses. So, less snails eaten by shrews. That was easy, wasn't it? Name given to the snails. What are the snails according to this list? What are the snails? Community, ecosystem, population, territory. What is the name given to all the snails in the garden? Well, they're not an ecosystem because that's the living and non-living factors. They're not a place, a territory. They are a population. Community would be all of the snails, all of the shrews, and all of the lettuce all together. Bish bash bosh. Right. Let me go to a slightly longer question. All right, someone was talking about Pete. Talking about Peter Piper picked a pepper. So explain how the destruction of peat bogs and the use of peat affects the temperature of the Earth's atmosphere. 4 marker. Oh my gosh, you can always watch it back tomorrow. I'll go through it all and then you can watch it back tomorrow. I will get there. Are you talking about the food security and all of that? I'll get there soon because I'm kind of whiffling through this. Yeah, nice. So really what we want to say is, I don't need to say this bit, but essentially peat is used in 2 things, right? It's used in fertilizer. This is not part of my answer, you do need to know it. It's used in fertilizer for gardens, right? Because it's full of minerals. And peat used as fuel. Now, oh yeah, I can say that. So yeah, we can say that. So when it is used as a fertilizer, let's go up here. Oh fudge, man, my pen is dying. Come on, pen, mate. When it's used as fertilizer, we want to say decomposes, releases CO2. Obviously, greenhouse gas. When I mention that's a greenhouse gas, could lick onto greenhouse gases. And then I want to say when it's used as fuel, burns, releases, combusted as a fuel. Why is it used as a fuel? It burns nice and slow, gives off a gentle heat. Fuel burns, releases CO2. Okay, and then I need to explain CO2, greenhouse gas, and I need to say global warming, right? Separate mark for global warming and greenhouse gas, global warming. And if I want, I could say traps the Earth's heat and all that business, biz. Okay, guys. Dead leaves contain carbon compounds and nitrogen compounds. Describe how carbon and nitrogen in compounds in the leaves are recycled and used by living trees. You should include a description of how the leaves are broken down, how substances are taken in and used by the trees. What a bummer. Nice. Yeah, who's Pete? That's what I want to know. Right, guys, you can talk about the carbon cycle here. Just remember, if this is... Fudge me. So you have something called the carbon cycle. So just remember with the carbon... Come on, pen. Move, pen. Right. Hold on, Tom. What you doing, mate? I don't think you've got to know a lot about the nitrogen cycle, so it's just really decomposition. There is a nitrogen cycle, but just decomposition. Let me run through the carbon cycle for you, for those of you that don't know the carbon cycle. So just in case you don't know the carbon cycle, it'll take me 1 minute. You've got trees. Trees take in CO2 from the atmosphere. They do that by photosynthesis. It's the only way that carbon dioxide is taken into plants. So in that answer, which I'll go back to in a second, we want to talk about carbon dioxide being absorbed by plants in photosynthesis. Now, also with the carbon cycle, trees release CO2 back in the air by respiration. And we want to talk about that a tree can die and decompose. And when a tree dies, skull and crossbones, you get the idea. Okay, that's the worst skull and crossbones ever. When the tree dies, the decomposers, which are bacteria and fungi, decomposition is really, they're going to do respiration. We call this decomposition. They release CO2 back in the air. So we've got respiration and decomposition putting CO2 in the air, and CO2 coming out of the air and going into living things as photosynthesis. What is the CO2 turned into in here? Well, it's turned into glucose and starch and protein. So you do need to know in the carbon cycle that protein, glucose and starch is all stored in the tree there. Then we have combustion, and combustion puts CO2 in the air there as well. What the hell is that? That's supposed to be a factory, but yeah, that is releasing CO2 by combustion into the air. So we have three things putting CO2 into the air: respiration, combustion and decomposition, and only photosynthesis taking carbon from the air and storing it in the body of the plant as glucose, starch or protein. Now you also have your animals that eat the plant, and then they die and decompose. Now, both of these, when they die and decompose, separately to all of this, we end up with nitrates in the soil. And that is from decomposition. We get minerals, nitrates in the soil, and they can get sucked up into the plant's roots by what? By active transport. So you might want to, when you go back there, you just want to say like, yeah, nitrogen from the air. Yeah, you don't really need to know a lot about the nitrogen cycle in AQA, so I don't really know. That question's a bit of a bogus question, but yeah, what was it? This one. Describe how substances. No, we don't want that one. Where were we? Yeah, so really, you're going to get probably 4 out of the 6 marks just talking about how carbon is recycled and used by living trees. So what I would say for this one, it's saying how is the carbon recycled from dead leaves. So my dying leaf, I want to say decomposition, and I'm going to smash marks for decomposition. I then want to say CO2 in the air, and then I want to say it goes back in the plant by photosynthesis. So by doing that, I'm talking about decomposition releases CO2 to the air. The CO2 is taken in by the plant leaves in photosynthesis. What is it turned into? It's turned into starch, protein, carbohydrates inside the plant. So I've smashed on how the carbon is recycled from dead leaves. Now, nitrogen in compounds in the leaves are recycled and used by living trees. Nitrogen, when the dead leaf dies, it releases nitrates. Now, nitrates is a mineral and it is absorbed by active transport into the roots of the leaf and incorporated with glucose to make proteins. There we go. Bosh. Right. What I want to do is get some other questions. Now, some people were saying, what stuff towards the end? Let me see what you mean by the end. Some people were saying, what's at the end of the topic? Let me have a little look. Oh, one thing that is almost guaranteed to come up is transects. So, yes, let me go through a little bit on your choice here. So, you're going to could well get a question with the dreaded quadrats, and this is a core practical for you. I'm going to take 2 minutes on this. That is it. So watch out for, if you see this word, I'm just going to say, if it's a 6 mark or a long quadrat question and it says population, normally if you want to work out the population, you take the field number 1. You're going to randomly, I'm not writing the full thing, you're randomly going to place 10 quadrats. Why? This is going to save time. Random avoids bias, and sampling, if you're asked why we sample, it saves time. Because I could just count everything in the field, but that's going to take me years. So randomly place quadrats. You can use your calculator random number generator, right? Calculate mean number in quadrat, right? So I'm going to add them up, divide by 5 if I've done 5, add them up, divide by 10. Find out the mean number of dandelions in your quadrat. What are you going to do next? I'm just going to write mole. You can work this out yourself. Multiply up to the size of the field, and that is your population. Now, how do you actually do that? Well, if I know this is 100 meters and this is 1 meter, I'm going to times my mean number by 100. So if there's 7 dandelions in a 1 meter quadrat and it's a 100 meter field, easy, 7 times 100, I've got 700 dandelions. Most likely the quadrat is not a meter squared, it's going to be 0.25 meter squared. So I take my 100 meter field, divide it by 0.25, and I realize I can fit 400 quadrats in the field. So I need to go 400 times my mean quadrat, 7 times 400, and I'll go, ooh, there's 2,800 dandelions in the field. So I'm just saying you've got to do the maths calculation to work out, given the area of your quadrat, what the number is in the field. That is the population version. Please watch out peeps, because the other version of this practical, they're going to use the word distribution or spread or changes, right? I mean, they might say changes in the other one, so I might be screwing you over here. But so what do I mean by this? If you're doing the distribution, you can tell from the style of the question. They're going to be like, oh, here's the beach, here's the forest, here's the lake, and we want to see the distribution of dandelions. They're not asking you distribution. Population means how many. Distribution means where do they live? Do they live near the lake? Do they live over here? Wherever. Do they live near the human footpath where humans are walking, or do they live near the lake? Where do the dandelions live? So what we're going to do here is we're going to place a rope or a tape measure. We call this a transect, and that is a rope, yeah, or tape measure. Better to be a tape measure, right? Because you've got distance on it. Then we're going to place quadrat at regular intervals. Not random. You're taking the quadrat and you're putting it 1 meter apart, because what you're trying to do is see, do more dandelions live near the path or live near the lake? So you're looking at distribution, the spread of the organism, not how many, right? So it's not random, although you could choose a random starting place, but that's going to overcomplicate it. So I just steer away from that. Place quadrat at regular intervals, and then you just need to say, count the number of organisms in the quadrat, 1 mark, identify them with your mobile phone, Google. So Google, use Google flipping identifier so that you know it's a dandelion. You can say that with the other one as well. And just remember, I didn't mention the word quadrat here. So what an absolute idiot. Randomly place quadrat. I mean, the word quadrat is a mark if they don't tell you. So they're the 2 different ways. You use a transect, measuring tape, rope, place the quadrat at regular intervals, and you do the same thing, count and identify organism. And look, explain how you'd identify it. You do not need to measure up by the size of the field here. You just plot them in a kite diagram or plot them in a graph. Okay, what topics do people want that are at the end of the trips? Melt practical. I already did a bit on DNA earlier. I did DNA earlier, so let me do the melt prac. Listen, I'm gonna do the melt practical so easily. Okay, I'm not gonna long it. I'm not gonna long the melt practical. Okay, can't be asked. So what you're gonna do? You're gonna get some milk. You're gonna put the milk in a tube. Milk's white. I want to know if it decomposes. When the milk breaks down, it has lipids inside milk. That's fat. Lipids are broken down by an enzyme. It's called lipase. They're going to be broken down into fatty acids, and they're going to be broken down into glycerol. Okay, now how could I tell that has happened? Well, the fatty acids, what do acids do? Acids lower pH for all you chemists. So what I can do is I can add an indicator that gives the milk a colour, and then when it goes acid it changes colour, and that I can use a timer. Time how quickly color change. So we're going to time how quickly the color change as a measure of rate of decay. Now I'm just going to check the notes on the old CGP. I believe they use phenylphthalein. Now phenolphthalein or phenolphthalein is a pink indicator, and so you start around pH 10. How do we create pH 10? We can use a buffer to create pH 10. So we can add sodium carbonate. Now sodium carbonate is alkali, so we add sodium carbonate. That is going to be an alkali, right? Alkalis are soluble bases. Sodium carbonate, most carbonates are insoluble, but sodium carbonate soluble. I'm going to add indicator. Now, if you can't remember, just call it indicator, but the name is phenol perphthalene. And I just think of it as phenol pH, like pH, phenol pH phthalene. So, and that is pink in alkali. Okay, so it's going to be pink at around, it's around pH 10. It's going to be pink when it's alkali, right? And I added the sodium carbonate. That's going to make the milk have a pH of 10. What's going to happen to the pH as acid is produced? The pH is going to drop. When it gets to pH 8, phenol, oh my good lord, perphthalene goes colorless. So what we can do, we can measure the rate, the speed of decay. You must have a stopwatch. So do remember you need a stopwatch like that with some timers, and you're going to need a stopwatch to time how quickly it goes pink to colorless. Now, is the milk going to go colorless? No, it's going to go white. So I said phenolphthalein goes colorless, so the milk is going to look white rather than colorless, right? It's not going to be transparent. So it's going to go white, but the indicator has gone from pink to white. Decay is finished. That can be my time to press stop on the stop clock. It doesn't mean decay is finished. It just is an indicator from when I can start and stop to measure how long did it take 5 centimeters of milk with a concentration of 5% lipase to change and make the phenolphthalein, 2 drops of phenolphthalein go pink. Now I need all those control variables. I need to make sure the amount of lipids is the same in the milk, so same type of milk. So your controls will be like, you're going to want like same volume, same conc. Now what of? I might be investigating lipase, but I'm going to say lipase enzyme. We want the same amount of lipase enzyme, same volume of milk, right? Same type of milk. I can't fit it all in, but you get the idea. So I want the same concentration of all that business. Now you can use a buffer as well. Sometimes they say use a buffer solution. I'll check that out. They probably say use a buffer here to maintain the pH at pH 10 until you start. And then the other thing is, what factors affect decay? So do you know the 3 factors that affect decay? You're going to change one as your independent variable and keep the others the same. So 3 factors for decay are going to be oxygen, temp. So you're probably going to get one where you're changing the temperature. Obviously high temperature you denature the enzyme, so you're not going to see it change color. It's going to stay pink, right? Because the lipase will denature and it can't make it go white, so it will stay pink because you don't get fatty acids, right? I guess oxygen, temp, and what was the other thing? Was it water? What do you need for decay? Oxygen, temperature. Was it? It was water, wasn't it? Yeah. I don't think they're going to give you. I think they'll do temperature. Let me check. If it's temperature, you use a water bath, or let's see what they use. I've got it somewhere. Oh, here it is. Here it is. 5 centimeters of milk, different test tubes, blah 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 blah. What are they investigating? You can investigate decay by observing the action of the enzyme lipase. Sample of milk that's been made alkaline. When the lipase breaks down the milk, the pH of the milk decreases because of the fatty acids. Okay. As 5 drops of, they use 5 drops of phenolphthalein. That's a control method. So 7 centimeters. I mean, you don't need to say 7 centimeters. You just keep the amount of sodium carbonate solution the same, the amount of phenolphthalein the same, which I didn't write. And then, okay. How long the color take? Repeat the experiment at a range of temperatures. Yeah, most of the time you're going to do temperature, and you can calculate the rate. The rate of decay is given by rate equals 1000 divided by the time. So the way they do it is the rate of decay is 1000 divided by time. What I didn't put under my controls here, guys, controls, you want to control the volume of indicator, phenolphthalein. Did I say volume of milk? Yeah, what else did I not say? I've forgotten what else I didn't say. they might do the whole thing of heat the lipase to 30 degrees and heat the lipids. Before you add them, heat them both to 30 degrees. Otherwise, the lipids might be at a different temperature than the lipase, and when you mix them, you don't really know the temperature. Okay. A buffer controls the pH of a solution. Okay, what else did people want me to go through? That was the decay practical. Let me have a little look here. Hold on. Biogas generators. Have you heard of biogas generators? Biogas generators, they take poo and wee, feces, waste, and they turn it into like methane gas, and they make biogas that we can use and burn as fuel. And as the bacteria feed on the waste, they produce a gas that we can then use as a fuel called biogas, and it's renewable. But there are two ways you can do this. You can do this with continuous generators or batch generators. Now, there's advantages and disadvantages to each. So a batch generator is I put a load of poo in a bucket, in a big container, in a big thing, and I let it generate the gas. And then I take that gas and I use it as a fuel, and then I wash it all out and I start again and I put all my ingredients back in and we make some more gas. So you do it in batches, in groups. This has an advantage. If it goes wrong and you make a poisonous waste, or it goes wrong, you only lose one batch. You haven't screwed the whole thing up, right? So you're like making your stuff one section at a time. The downside is there is a gap in between where you are not producing. So while I wash out the container and I have downtime, there is a day or so where I'm not producing any product to sell. If you use a continuous generator, you have got a big container and you're feeding poo in one end and you're tapping out the gas in the other end, and you never stop and clean it. It's just continuous. As long as you keep chucking in the poo and the gas keeps coming out, it's going at a continuous steady rate. This is large scale industrial process. However, obviously with a continuous, you get a downside, is that if something goes wrong with the system, then you're going to be producing a load of not good product. Anyway. What we got? So you do need to know about pollution. You need to know that water can be polluted by sewage and toxic chemicals, pesticides that run off of fields, insecticides and fertilizers can be washed into the river, and these can poison and kill animals that live in the river or the sea. So that can come from sewage, like waste from houses, like poo and wee basically, and it can come from chemicals from factories, toxic chemicals from nuclear power plants or factories, and they can kill fish and animals in the sea. You can also get plastics and litter that can get trapped in fish gills and interfere with marine life. But we also need to know that toxic chemicals and household waste and stuff can also be dumped in the sea from the land, and then from the air you've got sulfur dioxide, which causes acid rain, nitrogen dioxide, which cause respiratory problems in humans. Acid rain can change the pH of soil and water and kill fish and trees, and you've got carbon monoxide from cars and stuff that can interfere with hemoglobin's oxygen carrying ability. What else? Deforestation. You guys do need to know about deforestation, which is chopping down trees. So I'm just kind of rinsing through, making sure you know what deforestation is. Why do we chop trees down? Is it because humans are evil? No. Our populations are growing all the time, and we need to clear land for agriculture. Agriculture is a fancy name for farming, so you will get the same marks for saying farming. In order to grow more rice, more pasta, more bread, whatever, more wheat, more crops, carrots, potatoes, we need to clear forests to make space to plant. Okay, you'll be surprised, but a soccer pitch, the World Cup's coming up, a full-size soccer pitch can probably sustain 4 humans. If that was all carrots and potatoes, a whole football pitch, it will keep like just your family alive, no one else. So that's how much land is required to keep just 4 humans alive in terms of growing food. So Yeah, we have to clear forest. We also clear forest to get wood to build houses and wood to burn. Still 30% of the world heats their homes by burning wood. So there's loads of reasons for chopping trees down: to build houses, to make furniture, to make building materials, to use as a fuel, and to build farms to feed the growing population. Now, what are the issues? So they're the reasons why we do deforestation. Then what are the reasons, the bad results of deforestation? You destroy habitats, so we have less biodiversity, less species around. Some species become extinct. Looking at the carbon cycle, trees taking carbon dioxide. So if we chop the trees down, there's going to be more carbon dioxide in the atmosphere, more global warming, and all the bad consequences of those more greenhouse gases in the air. Okay. What else? Destroying peat bogs. Why do we chop up and destroy peat bogs? Peat bogs are used for fertilizer in the garden because they contain nitrates, phosphates, magnesium, an amazing amount of minerals, so your plants are going to grow wicked. However, when decomposers like bacteria and fungi break down the peat in your garden, carbon dioxide is released into the air, so it releases greenhouse gases. The other bad thing about destroying peat bogs is that lots of animals make that their home, and so various species will become extinct if we keep destroying the peat bogs. And peat is also used because it burns gently and gives off gentle heat, so it can create warmth and it lasts a long time, but burning it releases carbon dioxide as well. So we have to think about how to maintain biodiversity. Yeah, everybody needs sleep. Yeah, a buffer, Finn Singh. A buffer is a buffer controls the pH at the level you want. So if you want to start the pH at pH 10, you can add a buffer and it will fix it at pH 10. You might want to control the buffer. Karim Lashin, the use of transects. Transects are only when you're looking at a species and you're looking at the spread or distribution. There are like 4 bullet points. So you want to place a tape measure or a rope across a beach, a field, a forest, whatever area, a mountain pass, wherever you're doing your transect. It could be from your school playground to the school entrance. And what we're trying to see is, do more of our creature grow here or grow over by the school gate? And it might be that, people walking over the gate, they trample it, will expect less, but we need to test that. You are not trying to estimate the gram population size. You're not interested in the total population. That is the random quadrating, random sampling where you throw 10 quadrats. That is for estimating population size, comparing how many flowers grow on field A in the playground to field B in the playground. That is the random one. You do the random there, you do the random there, you multiply up by the size of the quadrat, size of the field. Transect, you're looking at how the species change, how the number of organisms changes from one side to another based on abiotic and biotic factors like the shade from the school building or whatever. So you're going to place a rope or a tape measure across the field. You're going to place your quadrats non-randomly. You're going to place them every meter at regular intervals, and you're going to identify using your mobile phone. You do it for both and count. And if it's a small organism like moss or grass, you have to estimate percentage cover. You don't count. It's too hard to count. Liver and kidneys. The grid box cast is the liver. In paper 2, what do you need to know about the liver? All you need to know about the liver for Bio AQA paper 2 is that the liver stores glucose as glycogen, and insulin takes glucose and puts it in the liver. Insulin in the liver, in sugar liver, puts the glucose in the liver, and there it is turned into glycogen. It stimulates that reaction. Now, glucagon, which is another hormone made by the pancreas, not the liver, glucagon is another chemical hormone that goes to the liver, and it takes the glycogen out the liver and turns it into glucose. Well, it makes the liver turn the glycogen into glucose, and then it takes the glucose out. So glucagon from the liver, glucose gone from the liver. Yeah, that's what the liver does in paper 2 if you're AQA. The kidneys are for excretion of urine. Protein is turned into ammonia and urea. Urea is a toxic chemical in our body that we must pee out of our body, and our kidneys do that for us. ADH is a chemical produced in the pituitary gland in the brain and travels by the blood down to our kidneys. And if I haven't had a drink, I haven't had a drink in ages. So my body is sending ADH down to my kidneys and telling my kidneys, do not wee out water. Take the water back and put it in the blood. When I go for a wee, it's dark, dark yellow and there's not a lot of wee coming out. Whereas if it's a winter day and I'm not sweating, I'm not using any water, and I've had quite a nice drink, my wee will look like water because ADH is not released from the pituitary gland and my kidneys are told, go for a wee. Let out lots of water. Yes, it makes the kidney tubules more permeable to water, ADH, so that more water is sucked back into your blood by osmosis. Oh my gosh. Eutrophication, Sabrina, is not something that I think you have to know on the AQA syllabus, but I'll go through eutrophication. Eutrophication is when sunlight, eutrophication is when farmers or even gardeners or anyone, the local council, when they water the plants, any fertilizer that they've put on the plants, any nutrients, right, like nitrates, phosphates, magnesium, when it rains, the rain washes it in underground streams and rivers down to the sea, down to lakes, down to the local river or the local stream, and it seeps through the mud into the water. Those minerals are not just needed by plants, but they're loved by a creature that lives in the water called algae. Algae, green algae, suck up all those nutrients and they bloom with a massive overpopulation, and the surface of your pond, your lake, your river goes completely green like a carpet living on it. And what that does, they block the sunlight from getting in the water. This kills the water plants like pondweed from the Pondweed Practical. It kills those, and those plants, remember plants photosynthesize, they give out oxygen. So if the algae blocks the sunlight and all the plants die, there's no oxygen in the water, and if there's no oxygen, then all the fish die because they can't respire. And that's the whole process is called eutrophication, and it's really caused by an algal bloom due to either fertilizers that farmers put on their fields or sewage, poo and wee from your house. If it flooded and poo and wee went into the sea and the lake, then the algae feed on the poo and the wee, the sewage, and they create an algal bloom. Block the sunlight and they toxify, the sterilize or toxify the water basically by removing all the oxygen. And as the algae die, because you live and you die, it's part of life, as algae live and die, bacteria feed on them, aerobic bacteria, and they use up the oxygen in the water as well and make it unlivable because there's no oxygen left. That is triple, but that's not necessarily AQA. Couple more things to do with AQA. Well, couple more things. Let's have a look at not really food chains. I don't really want to look at food chains, but I just want to remind you, if you're doing triple dipple, let's go like this. Let's say we have grass, rabbit, fox, right? Let's say we've got grass, rabbit, fox. This is called a pyramid of mass, biomass, or a pyramid of energy. They look exactly the same. Pyramid energy. Now, Einstein said, I could call it biomass because Einstein said mass equals energy, E equals mc squared. So he said that they're equivalent. So anyway, here's my grass. My grass might have 10,000. This is just, I'm not doing it accurately, 10,000 joules. Roughly 10% is the maximum that can go into the next level. So the rabbit can only get 1,000 joules of energy. So why can the rabbit only get 1,000 joules of energy? Well, there's some reasons. The rabbit excretes, it poos out some of the grass. It can't use those atoms. It can't use those atoms that it has pooed out. So it excretes. The rabbit, oh my gosh, I can't spell rabbit. Jeez Louise. Respires. The rabbit moves. Forget my spelling. I'm just going to put R. The rabbit moves, heats its body, right? A rabbit is an animal, a mammal. It creates heat. It has to do respiration. Some of that heat is to keep it warm. So maybe 1,000 joules were burnt off as heat to stay warm. Maybe it pooed out another 1,000 joules. Maybe it couldn't digest grass. Have you tried eating grass? The rabbit probably couldn't digest all of the grass. So some parts cannot be digested. So you get the idea. Now, you have to change these, excretion, and so you have to change these depending on the question. Now, the fox, how much can the fox get? Well, the fox can only get 10%. Guess what? The fox only gets 100 joules. This is the reason on the earth, if you weighed all of the grass, it will weigh 10 times more than all of the rabbits. If you weighed all of the rabbits, they will weigh 10 times more than all of the foxes. Because there just isn't, it's an impossible situation to have a heavier mass of lions than zebras. Because the lion eats the zebra, it only gets 10% of the zebra. So if you weighed all the zebras on an African plain, they would weigh 10 times more than the lions, because only 10% of energy is passed on. And this means our pyramid can only have a few levels. Maximum on land is about 5 levels with some food chains. For it to be sustainable. Now, in the water, it can be a longer food chain because algae are easily digested. They're a single-celled organism. There's no roots or anything. There's no hard bits that you can't digest. Algae are soft, single-celled. Same with mycoprotein as well, like if you look at like corn and stuff. Anywho, that is the reason why food chains are small, especially on land. Energy is wasted at each level. We call each one of these levels is known as a trophic level. Trophic just means energy level. Okay. Yes, you need to make sure that they are to scale. So if it says 10,000 joules, I'm going to make that 10 centimeters, but that wouldn't work, would it? Because then my rabbit would be 1 centimeter and my fox would be a millimeter. But you get the idea. You got to do 10%, 10%. You got to make it to scale and label it. Absolutement. You can do the central line. You can draw your pyramid in the central line. You can use the line to draw the two halves like that. You can do it like that. Okay, only 10% is passed on. You'll probably be asked why, and it is generally, but be careful you don't say the grass does a poo or a wee or excretion, and name the excretion. The rabbit will release feces and urine, so that is why it cannot, like all of the grass can't be put onto its body, all the energy cannot be incorporated. Now, as a farmer, I want all of, I'm going to say I'm selling rabbit. I'm a rabbit farmer. I want to sell my rabbits. These rabbits keep running about and burning off the grass they eat, the idiots. I'm going to lock them in a cage and not give them anywhere to move. Now you try and burn that off. I'm getting 20% of the food I'm paying for to feed them is going onto their body. When you buy a £1 chicken burger or a £3 chicken burger, how do you think the chickens have been kept? They've been locked in a cage because the person who's making those chickens, farming those chickens, doesn't want them running around, because if the chicken runs around, it burns off half of the food that I've just fed it. I want all of the food to become mass on the chicken so I make maximum profits and I can make 20 chicken burgers from it. So they lock them in a cage. It's called intensive farming. A little bit grim, and they inject them with antibiotics as well so that disease doesn't spread and that type of stuff. So yeah, cheap stuff. You're going to get cheap quality. But yeah, there we go. Anywho, yeah, fish farming, I'm going to put the fish in water, but I'm going to put them in big giant nets. So I could have a net the size of your bedroom now, or I could have a tank, and I could have it in a river so that they look, oh look, there's the river. But they're all inside this cage, and they're just swimming about. And yeah, I want to make the holes not big enough. I want to make the holes small enough so that they can't escape. And I might want to add minerals to the water or give them food. High protein, they're going to grow growth and repair, so I'm going to give them high protein, make lots of fish. But also when we're fishing, you need to be aware that there are laws on fishermen. When they use a net to catch fish, they have to leave big holes to let the mummy fish, they have to leave little holes to let, they don't care about mummy fish. They leave the little holes to let baby fish escape. And the logic is, I can eat mummy and daddy because mummy and daddy have had kids. If we eat the babies, they haven't had kids yet. So we're going to massively dent the population. So you only eat the fish once they've had babies, because then you've got the new generation of fish. And that logic means your grandchildren can eat fish and your great-grandchildren can eat fish because you're keeping the population going. So you make small holes in the nets to allow baby fish to escape. Yes, so what else? That's called food security. And you can also, as a government, make fishing quotas, which we have really strict fishing quotas in the UK. So the fishermen in the UK have to weigh their boat on the way out and weigh their boat on the way in, and then they work out the kilogram of fish they've caught, and they have to throw the rest back. Even if it's dead, they've got to throw it back. So they get really upset about that. But yeah, they have quotas to stop overfishing. You also got to know about biotech. So you do need to know about biotech. So if predator-prey cycle is dead easy, rabbit population goes up, so predator-prey cycle, you know that's dead easy. It's too easy, man. I probably shouldn't even bother going over it. But if the rabbit population goes up, so if that's rabbits, right, the fox population will follow after and start going up. As the fox population goes up, they're eating more rabbits, the rabbit goes back down. Guess what? The foxes get to a point, oh, there's no rabbits to eat, we're starving. They start dying. There's less foxes catching rabbits, so rabbits go up. And we call this predator-prey cycles. Now the foxes who are dying, suddenly there's rabbits everywhere. Oh, great, I can catch and eat rabbits. They start going up. So the predator always follows the prey in a predator-prey cycle. Anyway, water cycle, dead easy. All you got to know for the water cycle is you have the sea, you have evaporation, then you have condensation, and then you have precipitation, then you have runoff. You can just call that runoff. And then you have trees, which are also giving out by transpiration. And that's basically your water cycle. Lots of shons. 4 shons. Evaporation, transpiration, condensation, precipitation. Oh, and runoff. Anyway, yeah. What we want to do is get rid of that. And I just want to... Yeah, I should have really just done hundreds of questions. That's the problem. But what are you going to do? What are you going to do, mate? Blouse and skirt. Right. It's not always 10% increase. That's roughly what it is. It will be it's generally in the water. The water's a stable marine environment. You get a higher percentage increase than on land because us mammals, we've got to keep ourselves warm and stuff. The water is a stable temperature. It doesn't go hot and cold in the water like it does in the air. You know, if you get in the sea, it's always bloody cold. No, it's always roughly the same temperature regardless of hot or cold due to its high specific heat capacity. So, and also algae and plankton, which are the producers in the sea, don't have roots and stuff and undigestible bits. Bones are undigestible. So I haven't done the eye. I'll have a look at the eye. Can we go through bioleaching and phytomining? I don't think you need phytomining and bioleaching. That's chemistry, right? Yeah, I'll be doing a Chem 2 live if everything goes well, you know, if I'm not dead or something or whatever. Yeah, hopefully, yeah.
If you push the button,
Would you like it?
Like.
Do it. Do it.
Do it.
Let me just go through quick. Now, if you're doing AQA and you're doing triple, you need to know about genetic engineering very quickly. In fact, I can't even be bothered. But what do we do genetic engineering for? Genetic engineering, you're literally going to take some DNA and you're going to take a gene. This is a useful gene. What could a useful gene be? Maybe it's going to make my carrots resistant to herbicides, which are poisons that kill weeds. So this is a useful gene. Maybe it gives herbicide resistance, right? And I find this gene in potatoes. And I'm like, oh shit, man, I'm a carrot farmer. If only my carrots had that gene. Oh, what can I do? Well, I can use an enzyme and I can slice that gene out of that useful herbicide resistance gene. and then I can use more enzymes. Now, if you're doing IGCSE or something, you need to know the names of those enzymes, and they are restriction enzymes and ligase enzymes. So ligase, stick them in, and restriction, cut them. But if you're doing AQA, you just need to say enzymes, cut out the gene. So you're going to use enzymes, cut out useful gene. Remember, what is a gene? A gene codes for a protein. We did that earlier. Genes code for a special protein. So I want that gene because it codes for my herbicide resistance protein. So what am I going to do? I'm going to go, hey, where's my carrot? My carrot's over here. I'm going to take the carrot's DNA and I'm going to stick that gene in my carrot's DNA. Now, sometimes we do this via something called a vector, and a vector is a plasmid, a circle of DNA that will carry that gene for me. But we don't always have to do that. But you can use a vector. A vector is something that carries the DNA. This is now my transformed DNA. It has been transformed, right? It's been changed. Some people call it transgenic organism, but the animal that has it. Anyway, this is my transformed DNA. Or people can call it recombinant DNA. Some people in some exam boards call it recombinant DNA, call it transformed organism, transgenic organism, whatever. This is now going to get my carrot. How does this then help me? What, my carrot now doesn't get killed by herbicides. What's a herbicide? Herbicide is a chemical that kills weeds. So now my carrot, I can spray it with herbicides and it doesn't get killed. So I can pour herbicide on it. Why is that good? Herbicides will kill all the weeds, but not my carrot, and my carrot will survive. Woo! And that's called biotechnology, or GM, like genetic modification. Okay, now here's an issue. I am using GM carrots. Farmer Bob down the road doesn't want to use GM carrots. In fact, Farmer Bob is selling organic carrots. And he says to people, my carrots have not been messed with. They got no of this genetic engineering business going on. They're completely lovely natural carrots. Well, along comes a bumblebee and picks up some pollen, some sex cells, and then flies over here and drops it off there. Now what have we done? We've just brought a potato gene into the organic carrots. So even if you go and buy organic, and what does this mean? This means that wild populations can be contaminated with GM genes, and that's one concern people have. And they have concerns that, like, okay, so what if this herbicide resistance gets into the weeds? What if somehow the weeds get the herbicide resistance? Somehow they get that in them due to insects and business. So, yeah, you're going to end up with herbicide, and now the herbicides don't work and you can't kill the weeds. Anywho, I did the decay, I did decay. I will be doing physics. Someone said, can we do the eye? Eye. Let's have a quick look at the eye then. Quick look at the eye. I might go to bed after the eye. I think I might go to the bed. Go to the bed, my bed, a bed. Jeez Louise. Yeah, I think I will. I'm going to hit the hay in a minute, guys. I don't know whether I think it's better to do these things without teaching content, and better just to go through questions. Probably for the other ones, I'm just going to line up questions, like 6 markers. People want 6 markers in that, so I'll probably line up some 6 markers actually. For the chemistry 2 live and the chemistry paper physics 2 live, I'm just going to line up 6 markers. Anyway, this is the eyeball. You have something called the cornea. You have the retina. The retina is anywhere at the back here. This is the optic nerve. So we have a blind spot here. Can't get an image formed there. This is the kind of blind spot that everyone has in their eye. You can't get images formed there because that's where the electrical signal. And if, in case you're wondering, the optic nerve is a sensory neuron, because obviously receptors are in here. Receptor cells, and they're going to carry those electrical signals to the brain. That's going to go to the brain where we will interpret the image and coordinate the image. Now we've got a cornea. Now the cornea does protect the eye, but it also does bend the light a little bit. But then we've also got a lens right here. We've got a lens, and then we've got a muscle here. This is called the ciliary muscle. And then look, there's these little bits here. I don't know if you can see that. There's these bits here. These are suspensory ligaments. So them things there are suspensory ligaments. So what do they do? What does that do? Do. Now, if you look at something near, we call it accommodation. So what is accommodation? Looking at a near object. I don't know if I've got room. Let's have a little look. Looking. I missed out some bits as well. So we want to look at a near object. By the way, the colour bit of your eye is known as your iris. It's actually made of muscle. So it's all protein, that. It's all like made of muscle. People have different coloured eyes just because it refracts the light differently. They've all got the same muscle, but they have different amounts of it, and that refracts light differently. And then you have the pupil. The pupil is just a hole in your eye. You could stick a pin through that. Don't do that, you'll burst your cornea. But like literally, it's just a hole into your eye. And then you've got these blood vessels delivering oxygen for respiration to your eye. Now, when you're looking at a near object, I believe this is called accommodation. Accommodation or accommodation? I've got no idea. It's too late and I ain't had enough coffee. Looking at a near object, accommodation. So what happens when we look at a near object? We want the lens to go fat. So when you want the lens to go fat, when you're looking at near things, this is what happens. We're going to go ciliary muscles contract, ligaments loosen. I mean suspensory ligaments loosen. Lens gets fat, condenses, thickens, gets fat, whatever you want to say. Light focused on retina. So what I mean by that is when your lens gets fatter, the light is going to do this and focus on the exact spot you need on the retina to make an image. Okay? It gets more convex. So we want to say when we look at something near, the ciliary muscles contract, the suspensory ligaments loosen, the lens fattens, light gets focused on the retina. Guess what we would say if we were looking at something far away? We would do the opposite. Ciliary muscles relax, ligaments tighten, the lens goes thin, and the light focuses on the retina again. Now, how can you remember that? The way to remember it is when you're in bed and you're doom scrolling on your phone. I bet a lot of you sit there TikToking or whatever. What happens to your eyes? How do they feel when you're staring at your phone? Remember, that's near. How do your eyes feel? In fact, get a pen and do this. Stare at that for a while. See how your eyes feel. Read a book late at night. How do your eyes feel? They start to hurt. Why do they hurt? Because your muscles are contracting. And that means you build up lactic acid, and that hurts. So the way I remember this, what happens is I start with the muscles. I go, right, when I look at something close, my eyes hurt. That means that the muscles are contracting, making lactic acid and stuff, so they're contracting. That means the ligaments are doing the opposite. They're loose. And then the lens fattens and the light focuses on the retina. I kind of have to remember. But I can remember the muscle contracting and the suspensory ligaments loosening by remembering that when you look at stuff, it hurts. When you look far away, that's good for your eyes. So if you've been reading or watching a screen for ages, you should just go and stare off at something in the distance for 2 minutes. It relaxes the muscles in your eyes. So, yeah. When you look far away, the ciliary muscles relax. Suspensory ligaments do the opposite. They tighten. Now, muscles contract and relax. Ligaments tighten or loosen. You can't use contract and relax for ligaments. Anywho, that's that. Then we have this garbage. So You have myopia and hyperopia, whatever, can't even say it properly. But if you are long-sighted, so someone who is long-sighted, I'm long-sighted. This means what it says on the tin. You can see things far away. I've no problem with seeing things far away. Up close, things start to get blurry for me. Why is that? That means oh my God. My lens ain't fat enough. I know. My lens is too thin. Lens too thin. What does that mean? When I look at something close, the light rays don't have time to go like this. So you've got one or two options. Lens too thin or eyeball too thin. Either way, the light is converging theoretically behind my eye. So it meets the retina, they're still apart, and I see a blur. What's the way to correct this? Add a fat lens on your glasses. So my glasses are going to be convex, and they're going to bring the light rays in more and make it focus on the retina. So the key is that it focuses on my retina. Now, for someone who is short-sighted, they're great at seeing things up close. They ain't got a problem. But when they look far away, oh my gosh. Hold on, folks, one sec. I need to put my old specs on myself. It's getting tired. I'm getting tired. I need to get the names right here. One sec. I'm going after the light after I do the eyeballus. Maximus eyeball strain. I have to say, this is the bit of biology that I always get confused with. Right, what are we doing? Long-sighted, unable to focus on near objects, lenses the wrong shape, doesn't refract, bend the light enough. Did that. Light goes behind the retina. We need the convex lens. We did that. Right, short-sighted people. These are the people I'm dealing with now. They can't focus on distant objects. Yes, because the lens is the wrong shape. The eyeball's too long or the lens is too fat. So it's just the opposite. So short-sighted, they have an issue. Their lens is too fat, so that if it is a far away object, by the time it reaches here, it gets bent too much and it's falling before there. Now, short-sighted, their lens too fat. It might be that their eyeball's too fat. Either way, the light doesn't reach the retina. So you change that by using a concave lens. Concave looks a bit like that. It looks like a cave, like you're going in a cave. It dips in, so it's concave. What that does is that diverges the light like that, so that by the time it converges, it actually goes to the retina. But the main thing to focus on here is that the lens makes the light focus correctly on the retina. Now, there is one other thing which can confuse people. This is slightly separate. This is your pupil, and your pupil, if in dark light, our pupil gets bigger, and what we say is the radial muscles contract. The straight lines in your eye are the radial muscles. So if you look at your eye closely in the mirror, if you've got light colored eyes, it's easier to see, but you can see these straight lines like this in an eyeball. You can see them in the protein. They are the radial muscles. When they contract, they pull open the hole in your eye. Now, you have other muscles called circular muscles, and if you look at your eye in the mirror, if you've got kind of light colored eyes, it's easier to see. You'll see circles of shades of color in circles. These are the circular muscles. When you walk into bright light, they contract and they make the pupil smaller. The idea is to let less light in so it doesn't damage the retina cells. Remember, light is energy. Light has a wavelength, a frequency, so it can damage the cells. And then you'll get a question like this. Describe the changes in the pupils and the iris going from A to B. Explain how these changes occur. Refer to the changes in light level in your answer. So we'd just be like, yeah, the person has gone into, so they've gone into bright light. And what's happened? Well, we would say the radial muscles contract. That means the circular muscles are relaxing. The radial muscles contract. the pupil gets bigger and more light is let in so they can see in dim light. You get the cue. Okay, and then there's one like this. Explain how the blurred vision is corrected. We can see here they're using some sort of concave lens. So the light is falling short. What do we need to do? We need to extend the light so that it gets there. I don't want to make that fatter, otherwise it's going to go even shorter. I want to extend the light, diverge it out. So what we're going to do, we're going to put a concave lens. So instead of the light going like that, hitting there and going in, notice what this lens does. This lens takes the light and it sends it out like that. So now the lens is bringing it back in, and look, if we get the thing right, it fits perfectly on the retina. So we want to end up saying light, whatever happens, light, this is always going to be one, light focuses on the retina. Okay, so I want that. I know whatever my answer, that's going to get me a mark. Explain how the blurred vision is corrected. This is a concave lens. So I'm going to say we're going to use a concave lens. If I want, I call it a diverging lens. It diverges the light rays. So that diverges the light rays, and then the light focuses on the retina correctly. Yeah, that will do me. They didn't say explain why we have blurred vision. If we had to explain why we have blurred vision, the person's eyeball's too big or their lens is too fat. So the light rays are coming in too much. So whatever. Yeah. All right, I'm off, folks. Convex lens is for people like me that are long-sighted. Yes, so I can see far, but I can't see near. You put a convex to make it fat and diverge it onto the retina. I'm off. All right, guys, take care. Yeah, it's been, you know what, for the next one I'm not going to do this for C2 and P2. I'm just going to do questions, because I think this style is too long to get through the course. I think it's better to just smash it out with the questions. I have got a live I did yesterday which was 100 B2 questions, which kind of like, you can cover the course and more of the course quickly and in a more engaging way with questions, I think. So all right, guys, take care. And yeah, smash the exams. Remember, there are always other channels. Don't worry, there's other channels. There's other channels, people. There's other people. So don't worry. I'm going to be off. Yes, brothers and sisters, welcome to exam.com. Now there's 32 seconds left before the last exam of this exam season. I know what you're thinking. Is it too late to invest in our incredible slides which cover all of the content? Of course it's not too late, man. There's 29 seconds left before the final exam, and just for 999.99999, you can get our incredible PowerPoint slides. Just check out the quality of these slides. Look at them. All right, now I know what you're thinking. Am I doing this just for the money? Well, I'm not. This is to help you, because I want to help you. Yeah, that's what we're here for. For just 999.999. I know what you're thinking. We're now 2 minutes into the last exam. Oh my gosh, is it too late to get this? No. Put your hand up. Ask the invigilator if you can go for a wee. Pull out your phone and just get these slides right now. Oh, and by the way, if we get 50,000 likes tonight, then Mo's going to get his hands out.
You're still here? It's over.